Good morning. So, how are you? So, we'll start our rapid revision or marks booster, and we'll focus on some important area, right? We won't be able to cover the entire length, but then we can focus on some prominent things which are there chapter wise, and in that way, we can start our discussion with the important topic related to biotech, right? So, biotech is again a lengthy area, so we'll cover it first, and one by one we will select few topics and we'll talk about it. So the first topic in biotech that we should talk about is genomics, right? So genomics is the first topic we are going to discuss, and you know that genomics is a study of genome. And genome is nothing but the total gene present in a particular organism, right? So total gene in an organism, right? So all the gene in a one go, if you want to say, you can call genome, and a study of genome is known as genomics. Now there are many things related to genomics. There are two things that we can talk about in genomics. One terminology which is important nowadays, that is called metagenomics, right? And the second terminology, that is called personal genomics. Personal genomics. And another term is genomics itself. So whenever we say genomics, genomics means that you get a sample of population, right? Whenever you talk about genomics, it's not about single individual. It's about many individual, right, of a particular species. Maybe 10 people, 100 people, 1000. The sample size will vary, right? So let's say when first genomic study started, Human Genome Project, 1000 sample was collected from human beings. It was a human genomic study, so it was not for single person, it was for group of person, right? So genomics is always about that you study genome of a population. So here we can understand that in genomics what will happen, the study of genome of a population group, right? So there it won't be like one particular organism, it will be always a group. You know that definition of population is that population is always belongs to single species, right? So one species, and then you get the sample size population group. Metagenomics is something else. Whenever we talk about metagenomics, it is about community. You know, in environment we talk about population and community. Population is always about single individual, single species living in an area. Community is many population living in a particular area. So whenever we talk about genomics at community level, then it is called metagenomics, especially microbial life, right? So it is metagenomics is what? Study of genome at community level. Whenever we say community level, then it will be having more than one species. So many species. Many species. Whereas in genomics, normally when we talk about population, it's a single species, right? And many species, especially we use this term for microbial life. So microbial life, microbes, the microorganism, although we can take it beyond microorganism also. Let's say there is something called aquatic metagenomics. You get the sample of water from any lake or from any water body, and whatever organism which are present in that sample of water, you try to study the genetics of that. It may be several kind of bacteria, then protozoa, have fungus, algae, all the things put together. When you study the genome of it, then it's called metagenomics. similarly, I mean, UPSC is asked question from metagenomics, aerial metagenomics, right? Similarly, aerial metagenomics is one area. Soil metagenomics can be another area. You just take sample of the soil and whatever organism present in the soil, that you study the genome of that, right? Idea about in a particular area, right? You also know that what need to be done, what kind of species are living in that particular environment, and what is the relationship between two species out there. Personal genomics is emerging area, and this area is all about genome study or genomics of an individual. So individual, right? One person. When I say individual, it's one person, right? The genome study of one person, that will be known as permanent, because this will also help in, that is called personalized medicine. Personalized medicine, right? Nowadays, we are going through the process of personalized medicine or custom drug, right? Personalized treatment and all kind of thing. And when we go through personal genomics, then there is another aspect of it that is called risk assessment. Once you look into your genome, then you find out what kind of disease that might occur in the future. So that is personal medicine, custom drug, and risk assessment. Risk assessment. So genomics we can divide into three parts: normal genomics, population level; metagenomics, community level; and then, normal genomics, population level; metagenomics, community level; and the personal genomics, that is individual level, right? Now, if you go with the genomic study, so what we have done in genome, so we know that first we have done human genome project. Human genome project, right? It was done 1990, 2003. It was done already, right? And it was a first of its own kind. Human genome was just studied entirely for the first time. USA initiated it. Second thing that we started is called Human Genome Project, right? Human Genome Project, right? So what was Human Genome Project? First one. The idea was to take the DNA sample from the human being, read all the information and transfer it to computer, so that you can, in future, you can analyze it, you can find all the clues from genomic studies. But in Human Genome Project, right, what was there, that we created the entire database on computer, and then we imprinted this database on a artificial human genome. So here it was that you created something on computer, and then this data was transferred to the DNA that was made in lab. So basically it has given you artificial human genome. Artificial human genome. So this was we read the information from the actual DNA of the human being. In Human Genome Project, right, artificial human genome was created by predetermined data on the computer, right? So it was that. But recently, we started another project that is called Earth Biogenome Project. Earth Biogenome Project, right? 2018, and it will go till 2028. India is part of it. And in Earth Biogenome Project, what we are doing, we are sequencing genome of, that means studying genome of all living creatures or living organism around us. So 15 lakh living organism which are living around us, we are going to map it. And we are in that process. We have done many things out there. Because it's Earth Biogenome Project, that means entire Earth biodiversity will be mapped. Every country need to contribute some. So here, India is part of Earth Biogenome Project. Right. So India is participating into it. So these three things which are at international level. So one, we have done it. This is also going on 2016 to 2026 going on. Maybe next year we are going to get more details from this project. This is also going on. And Indian contribution is there because in Western Ghat we have many endemic species and we are mapping all of them. What India has done, because these are international projects, so the most important part in genomics is Genome India project. India project that we call GIP. So in our regular classes, we have already studied that when this Human Genome Project was completed, then India got the data from USA so that we can study it. But we could find that USA data is not very good for Indian population, right? Because our representation into International Human Genome Project was not there, not sufficiently there. We wanted to study the genome of genetics of Indian population, right? India is very diverse ethnic group and people are living out there. And our genetics is a bit different from European and American Western genetics. Obviously, when we live in a particular environment, an environment will have its impact onto our body and genetics in a long run, right? For example, if I can give you that in Western population, cancer is problem out there, but there the skin cancer is the major problem, right? They are basically doing more research into skin cancer. Whereas when you come to Indian population, skin cancer is not that important because the cases are very less. In our population, oral cancer, lung cancer, throat cancer, these kind of cancers are more important in terms of study and research because the cases are very high, right? For that matter, India is turning into and turned into diabetes capital. You know that. So we have to look into that, right? So genomics, in a very first way, it's all about that studying the human genetics to find out the diagnosis, the cause of the disease, the genetic basis of the disease, and then ultimately to get the better treatment, right? So what we have done, then we started Genome India project, and Genome India project is all about Indian population so that we can have it, right? तो पॉइंट आप लिख सकते हैं 20 in 2020 Department of Biotechnology started Genome India project. In this project, DNA data of 10,000 Indians. DNA data of 10,000 Indians are being studied at genomic level. Right. Aim of this project is to decode genetic diversity of Indian population and different ethnic group. Indian population and different ethnic group. Right. So it was in news because in January 2025, the first set of data was released, right? And PM Modi, he released the data, and the data is stored at whenever we study all these things, then we collect the data, and data are kept inside the computer. They can anyway. So you can write next in January 2025. January 2025, Genome India database was released. And it is available to researchers across the world. Researchers across the world. This data is stored. This data is stored at Indian Biological Data Center. At Indian Biological Data Center, Faridabad. Right. So this is what we have done. 10,000 individual we wanted to map. More than 5,000 we have mapped. More than 5,000 we have mapped, right? And the first phase of data is already available. And in a coming time, we'll release more data. So that is one current event that happened. And not only current, it's a static also. Because in 2020 we started. And the data is released in 2025. The next phase of data will complete this entire project. The second thing we should be knowing about genomics is genomic surveillance. So there is something called genomic surveillance. Genomic surveillance, the term is important. So genomics is not only limited to human beings. Genomics is also being used to have a surveillance on different variants of the disease. Right. Two diseases we'll talk about. One is corona. And you know that corona variants are there. Right. And variants of corona virus can be detected only when you study the RNA of the coronavirus. I mean, coronavirus is RNA virus. Their genetic material is RNA. And if you want to know that a particular coronavirus is a new coronavirus, a new variant or old one, then there is only one way. And the thing is that you have to go with the genomics of it. Genomics is also known as genome sequencing or gene sequencing. So genomic surveillance is that only. So whenever we come across any disease and we have a fear that this disease might have a new variant of the pathogen, the only one way to detect it, to get inside the genetics of the disease causing pathogen and then study the genome and find out. So for that, a project is already going for corona, that is called INSA-COG, and now we are planning the same thing for tuberculosis, TB, because in India TB is having different type, normal TB, MDR TB, XDR TB, TDR TB, right? Right? This is a study of genome. This is a study of genome of pathogen to find out new variants of that pathogen. new variants of that pathogen. It is done by genome sequencing. It is done by genome sequencing, or it is done by genome sequencing, or by gene sequencing. So both terms are used interchangeably. It's the same thing. Right. So this is what the genome sequencing, and for that we are having INSA-CoG. So INSA-CoG. INSA-CoG is right. So that is for COVID management, right? Because sometime you see that this COVID variants can come up. So that is Indian SARS-CoV-2 consortium for genomics. So Indian SARS-CoV-2 consortium for genomics. Right. INSA-CoG. So INSA-CoG is nothing, it's a chain of lab. It's a chain of labs to detect new variants of COVID. Right. And same thing we are going to do in future with TB. Right. So INSA-CoG is already working, and you can write one more point. Similar genomic surveillance program, is being developed to monitor variants of tuberculosis, TB, variant of tuberculosis in India. Variant of tuberculosis in India, right? So this is one important thing you should be knowing. Now, if we talk in that sphere of genomics, then there is something called eDNA, which was in news, and it's an important concept now. eDNA means environmental DNA, right? The DNA that we can collect from the environment, which may belong to different species living in that particular area. Some of the species are visible, you can see them. Some of the species are not visible because we are not able to monitor them. The study of eDNA is a kind of metagenomics only, and it is also a way to find out the biodiversity of a particular area. It will also help you to find out the migratory route, right? That how an animal or birds has migrated and whether they visited a particular place or not. Because when you look at any area of India, any wild area, or let's say national park, wildlife sanctuaries, then there are people who are always monitoring the species inhabited over there and a species living out there and a species visiting that places. So we try to record it with the camera, sound and all kind of thing. But many a time it's not visible. So what they do, they collect the DNA samples from the area itself. And DNA samples can be coming from the blood, feathers, bones, dead bodies, right? Even fecal matters, the blood droppings, right? And if you find that DNA, then it will give you a direct evidence that this particular place has been visited by those species. So you can write eDNA is present in environment. eDNA is present in environment due to life activities of living beings. Life activities of living beings. In forms of egg, sperm, excretory waste, blood, bones, feather, dead bodies, etc. Right? Anything that may be present. And you can write one more point. It is helpful in monitoring and conservation of. It is helpful in monitoring and conservation of biodiversity, monitoring and conservation of biodiversity, and protection of, and in protection of migratory route of animals and birds. In animals and birds, right? So that's the concept of eDNA. Another thing we'll talk about is cfDNA. That was also covered extensively. That is called cell-free DNA, right? So cell-free DNA, the concept is very simple. In our normal cell, we have DNA at two places. So one is called nuclear DNA. This is your nuclear DNA. And mitochondria will also have DNA. So that is called mDNA, mitochondrial DNA. So one thing we should be clear, that in our cell, in human cell, we have DNA at two places, nuclear DNA and mDNA. But because these are present in a boundary of cell, contained in the cell, that is called cell DNA. So we can call it both of them cell DNA. But when the cell is dead, so this cell will be fragmentized, and then DNA content of all these things, let's say this is having DNA. So this DNA content will come out, right? So after death of the cell will fragmentize and DNA content will be there in blood or blood plasma. So these are the DNA out of the cell, inside the blood plasma or blood, then it is known as cell-free DNA. The DNA which is free from the boundary of the cell. And we all have it. I mean, every animals or every human beings that we talk about, we do have cell-free DNA because cells will keep on dying, right? Death of the cell in the body is a normal thing. After a particular period of time, the cell will die, right? So you can write a point. cfDNA are fragments of DNA. Fragments of DNA present outside the cell, structure, in blood or body fluid. In blood or body fluid, like plasma or lymph. Blood or body fluid like plasma and lymph. Next point, it get released when the cells are dead and fragmentized. When the cells are dead and fragmentized. Correct. So that is cfDNA, and this cfDNA can have three prominent application, right? So first application, that means this cfDNA are fragments of DNA present in the blood and coming from anywhere in the body. Wherever the cell is dying, the cell is getting fragmentized, then DNA will be released. There are different forms of cancers that can be detected by studying the DNA molecule, right? And for that, we need to take the sample from the tumor site only. So cancer tumor, you take the sample, you take the cell of the cancer, you take it to the lab, study the DNA, and then you come up with the confirmation whether the cancer is there or not. But nowadays, we don't need to do that. Let's say the cancer tumor is there in lung. We don't need to reach to the lung tissue to get the sample out from there. What we can do, we can just take the sample of the blood. In the blood, you will have cfDNA of that particular tumor. Because in cancer cells, tumors are there, and tumor cells will be dying also. So when the cancer cell dies and gets fragmentized, then the DNA will be released into the blood. So collecting the blood sample from the patient can tell you whether the cancer is there or not, right? So little less invasion, still we can detect the cancer in an easy way. So the one area that we are using for detection of cancer, and especially we have developed one technology that is called Gemini test for lung cancer. So in Gemini test of lung cancer, we don't need to take the sample from lung tumor. Rather, we just collect the blood sample. In the blood, we can get the DNA, and this DNA is coming out of the cancer tumor in the lungs, right? Second one is called NIPT, non-invasive prenatal testing. So that is called non-invasive prenatal testing. That means if you want to test the genetics of a baby living inside the mother womb, then the older technology is amniocentesis, where we collect some sample from around the baby by putting a long syringe into the mother's womb. You take sample from the womb itself, from the baby's area. But then it is invasive method which may lead to miscarriage in many conditions. It may lead to some damage to the baby also. So now people are preferring for NIPT. NIPT means the baby's cell which came out of the baby's will die, get fragmentized, and it will come into mother's blood. So mother's blood will also be having the fragmented DNA of the baby, cfDNA of the baby. So once you collect the sample of the mother's blood, there you can find two types of cfDNA. One is mother cfDNA, another is baby cfDNA. And now analyzing baby's cfDNA can give you the information about genetics of the baby. You can rule out any kind of genetic disease that might occur, right? So that is called NIPT, and it is more. So this is used to detect genetics of baby, or let's say fetus, not baby, rather we can call it fetus, because that is still inside the mother womb, right? Yes, please. Yeah, it's effective, very much. Good. No, big tumors are uncontrolled cell division, right? And tumors have tendency, I mean, tumors have lump of the cell. They will die eventually, and fragments would come out. So this is a good way, because that is why it has been allowed. Otherwise, we could not have allowed it, right? The third area of cfDNA, that we monitor the patients who are undergoing any kind of organ transplant, right? So if organ has been transplanted in a patient, then this organ might get rejected into the body, because WBC will fighting against this organ. WBC has a simple rule that anything from outside, WBC will start fighting against it. But for a successful organ transplantation, WBC should not fight. So what we do, what doctors does, they put the patient on some medicine that is called immunosuppressant. So when the patient has gone through any transplant, let's say kidney transplant, then that patient is put under immunosuppressant medicines. So the WBC's activity will be suppressed, right? And then they will keep on monitoring the blood samples also. If they could find that some cfDNA is coming from the kidney, it means that kidney cells are dying. Let's say you get 1% of cfDNA from kidney. It means what? The damage is almost 1%. But tomorrow you are getting 10% of the cfDNA from kidney, because kidney belongs to somebody else. So that DNA will be different from the person's DNA, patient's DNA. So the magnitude of the DNA, cfDNA coming out of the kidney can tell you whether the things are going to be accepted or rejected. So it is for monitoring the patient going through organ transplant. So you can write for monitoring the patient going through organ transplant. Right? So this area that you should cover in genomics, right? Although very extensive area, but again the focus should be the overall human genome, what we have done, Human Genome Project read, write, that initiated at international level, Earth Biogenome Project, that is entire living creature living around us. Then come to Indian part, and the most recent development is GIP, and the data has been released, right? Next topic we are going to discuss is GM crops. Again, it's evergreen topic, right? And we'll focus on some important area only. So GM crops, here two types of GM crop we can talk about in India. One is called transgenic GM crop, right? Another is called non-transgenic GM crop. Non-transgenic GM crops. So transgenic and non-transgenic, right? All the examples that we go through, these are nothing but the transgenic one. In transgenic, what is there? Transfer of a new gene would be there. Addition of a new gene will be done. So what you are going to do, that you are adding a new gene, right? Or outside gene, foreign gene you can call it. So adding a new gene, outside gene, so that you can get some benefit out of it. That is called transgenic GM crop. Examples are all of them that we have studied, like BT cotton, right? Or let's say, BT brinjal, golden rice. These all are transgenic only, right? Non-transgenic is something where we don't add any gene, we just remove the gene out of the genome. So here what is the approach? Removal of gene from plant genome. Obviously, we know about the GM crop, that why do we need GM crop? We need GM crop so that we can get some new character into the plant, and that can be achieved by two ways. One is transgenic way, that is adding a new gene by genetic engineering, and another one, that removal of some gene, some unnecessary gene out of the crop genome, so that we can make the crop in our own way, right? Because obviously plant genome is according to plant's life, and we sometimes try to eliminate some of the character of the plant so that we can make it more useful in terms of our own agricultural application. The one example I can give you, let's say plant will have its own lifespan. Let's say rice, wheat, or any plant will have its own lifespan. So earlier the rice or wheat 4 months long, 5 months long, but now we have reduced lifespan. Maturity period has been reduced by different ways, hybridization and all. We have not done this particular thing, but what I am suggesting, that if a particular plant is having a longer lifespan, it is going to give you fruits or let's say product after 1 year or 8 months, then we can remove some genes out of it and then we can reduce the maturity period. Now we can make this plant to get mature only in 6 months or maybe in 5 months, right? Sometimes plant's height is too much and plant will start bearing fruits unless it reaches to a particular height. Now we can manage that too. The plant height is normal and still it is bearing fruits. So such kind of things can be done by the non-transgenic and obviously in a transgenic way also. So India is experimenting with both. This is a new technology. This is something that we are already using it, right? The most important thing you should be knowing about it, that risk factor is a little high over here, so that you understand, because you are adding a new gene, so the risk would be a little higher. If risk is higher, the risk assessment would be done for the longer period. So this kind of crops which are transgenic in nature, where the foreign genes are being added, will be having more risk towards the environment, human health and other way. It will require a longer risk assessment period, longer field trial, and it will be a little slow to get accepted into the agriculture. Yeah. We have just accepted only one, and that is what BT cotton. Rest of the things are still under risk assessment or let's say field trials. Here what will happen? Accordingly, if you talk about, then risk factor is less because you are not adding any gene into it. You are just removing the gene out of the genome. So risk is less. It means what? Acceptance would be faster, right? One more thing that we should be knowing, that many countries are not yet open for transgenic. Europe is not using transgenic. They are not accepting transgenic. We are one such country who export a lot of things to the European countries, right? And they are close for transgenic products, right? But they don't have any problem with the non-transgenic, right? If we talk about European countries, they are not against genetically modified food. They are against transgenics. There is a difference between transgenic and modification. Modification are both. These both are genetic modification, gene modification, right? But this is modification done by adding some foreign gene, which is having more risk. So it is not being accepted. So you can write European countries, you can add just one point. European countries are close for transgenic crops. Transgenic crop. And here you can write European country can accept non-transgenic GM crops. Non-transgenic GM crops. Right. Now, what kind of benefit we are expecting from it? So, if we go with the benefits of GM crops, genetically modified crops, including both transgenic and non-transgenic. So, first benefit, we talk about 4 major benefits. We'll focus on the major benefit only. One is increase in productivity. Increase in productivity. That's the first one. And the best example that we can give from Indian point of view is GM rubber. GM rubber. So we have developed GM rubber, and India is the first country to develop GM rubber. Even the first GM rubber plant was planted in Assam, and that was first in the world itself. So GM rubber is all about that developing a rubber variety which can grow in cold climatic condition of northeastern states, right? Because rubber is coming from Amazon forest, so they are habituated to, they're originally belonging to the places which are hot and humid. So rubber are acclimatized to, it is adapted to hot and humid climatic condition. That is why the southern part of India is able to produce good amount of rubber. But Northeast is also producing good amount of rubber in spite of having 4 months of winter. So we wanted to increase the production further in northeastern states. And for that, we wanted to make one change. And the change is that make the rubber suitable for even cold climatic conditions, right? Because in northeastern states, humid condition would be there. Rainfall will be there all through the year. But 4 months of winter will not be able to produce good amount of rubber. So we have done it. So you can write just one point. GM rubber can give good production in cold climatic condition of northeastern states. Cold climatic conditions of northeastern states. Right. GM rubber has been developed by Rubber Research Institute. Rubber Research Institute of India. Right. World's first GM rubber. World's first GM rubber is planted in Guwahati, Assam. Is planted in Guwahati, Assam. Right? So that is what we need to know. Indian context will focus more onto that. Obviously, we have to cover a lot many things. Second thing that we can talk about, the second benefit, that is how we can provide better nutrition, right? Right. So second one is better nutrition. So we can play with the genes to get the better nutrition, and the best example of this is golden rice. Golden rice. Right. Golden rice is also in news. All through the time it was in news, and it is in news because Philippines became the first country to accept the golden rice for commercial cultivation 3 years back, and only country who is growing it. But still in Philippines also there is a lot of controversy going on. वहाँ भी legal hassle चल रहा है, right? So what we have done with the rice, that we have added the gene of beta carotene with rice, and then this rice will not only producing starch, but it will also give you vitamin A. So you can write just one point. In golden rice, beta carotene gene has been added. To produce carotene pigment. Carotene is a pigment. Full stop. This pigment, this carotene, converts into vitamin A inside human body. So remember, this golden rice is not having vitamin A. This is having beta carotene or carotene pigment. It's a yellow pigment, right? Orange reddish pigment. So once you consume this beta carotene will convert into vitamin A inside the human body, right? Next you can write, golden rice was developed to counter vitamin A deficiency. Next point, in 2021, Philippines became first country in the world. Philippines became first country in the world to accept golden rice. To accept golden rice for commercial cultivation. To accept golden rice for commercial cultivation. Right. So now, next one, golden rice. एक इंटरनेशनल प्रोजेक्ट था, आई मीन जिसमें रॉकफेलर फाउंडेशन यूएसए along with two scientists Ingo Portricus and Peter Bear on a humanitarian ground they have developed it. It's not a kind of one nation project. It was a humanitarian project funded by a foundation, right? इसलिए इसमें पैसे नहीं लगेंगे. This technology will be freely available to all the countries who want to use it, because the motive was to counter the vitamin A deficiency across the world, and especially vitamin A deficiency are there at low income group, है ना? तो उसके लिए था. एशिया और अफ्रीका में ज्यादा है, एंड दिस टू पर्टिकुलर टू कॉन्टिनेंट दे आर राइस ईटिंग कॉन्टिनेंट्स, तो इनकी इंपैक्ट भी इधर ज्यादा होगी, राइट? सुपर बनाना ह्यूमैनिटेरियन नहीं है, वो तो है कि ठीक है, उसमें भी हमने बीटा कैरोटीन और प्रोटीन बढ़ा दिया है, बट देन सुपर बनाना इज अ काइंड ऑफ एक्सपेरिमेंट दैट वी हैव डन, बट एक्सेप्ट नहीं किया कहीं भी, है ना? Because obviously we have to go with the basic things first, right? The next one we can talk about is herbicide tolerance, third one. Rather, this area is now very important because of particular one change. The change is that when India developed, herbicide tolerance, we have one example, and that is what? DMH11, right? So Dhara mustard hybrid 11, it's nothing but the GM mustard, right? So many people are there in YouTube also and online also. Just I want to make one clarification, because I am going in a rapid mode, so it might not make sense if you have not gone through the entire basic thing, है ना? तो फिर लगेगा कि सब कुछ ऊपर ऊपर ही चल रहा है. So it's a kind of cake is already been made, we are doing just icing, right? So if somebody is fresh into this, तो उनको लगेगा कि सिर्फ आइसिंग ही है, केक तो है ही नहीं. So for that you have to go through the basics first, right? But I'm sure that because जो भी लोग उनको पता ही है, तो 95% लोग ऐसे ही होते हैं जो पहले बेसिक्स पढ़ चुके होते हैं. 5% आ जाते हैं क्योंकि ओपन है. तो प्लीज दोज 5% पीपल, आई मीन दिस इज नॉट द एंटायर स्टोरी. देयर हैज बीन अभी तक 1 घंटे में जितना पढ़ाया वो 10 घंटे में पढ़ाया था, है ना? तो 10 आवर्स इन अ क्लासरूम, 1 घंटा अभी, तो बहुत डिफरेंस होता है, राइट? So it's the whole idea is that picking some important things, then presenting in front of you, so that recall process is there, है ना? And if there is any addition, I can focus on that. Anyway, so DMH11 GM mustard. So it is HT variety. It was HT variety, लेकिन अभी focus shift हो गया, है ना? Focus shift हो गया कि now we are having this because of the Barnes-Barestar system, right? So here what we have done in DMH11, we have added 3 genes. So 3 genes has been added. One is Bar gene, then Barnase gene, and there is Barestar gene. Barestar gene. Now here the Bar gene is for HT. This is for HT. But now this Bar gene is not very important. HT is something that we are not talking now, right? This Barnase and Barestar, these 2 genes are important for cross-pollination in self-pollinating crops. So those crops who are self-pollinating, or let's say bisexual flower, so there we can use Barnage and Baristar, right? So the real focus is here now. So we started this DMH11 as a HT variety. That HT variety will be there, and we will be able to manage the weeds in a proper way by sprinkling the chemicals. But now the things have changed, and these things changed because of the long legal battle going in Supreme Court after PIL and everything. And now the scientists who are promoting it, or government who is promoting this technology, they are saying that HT is additional thing. It's not that necessary one, right? But having this particular Barnage and Baristar system is something which is important, because there are many crop varieties which are self-pollinating, and we want to cross-pollinate them, even for hybridization, right? Not for genetic engineering, hybridization only. Then also this Barnage Baristar genes could be very useful. So the focus over here, right, two things. One is HT, herbicide tolerance. That means you can use the chemical herbicide to manage the weeds properly into the crop field, in mustard crop especially. But then this has become secondary. The primary concern and primary interest is lying with Barnage Baristar system. तो ये पूरा सिस्टम को क्या क्या दिया हमने, कहा बार्नेज बारिस्टार system. So remember this, and this will allow you to develop cross-pollinated crop varieties if the plant are predominantly self-pollinated, right? And you know that the status of DMH11, recent status, that environmental release has been done. Government was all set to go for the commercial release in 2025. But then just after environmental release, PIL was there in Supreme Court, and case is going on. Supreme Court has already ordered that let's go with the existing testings and all, so that we can rule out any kind of adverse possibilities, and then only we'll decide what to do. That means the entire thing is in dilemma. Nothing, no conclusive verdict has yet come, right? So you can just add one point. Environmental release of DMH11 was done. Environmental release of DMH11 was done, and it is waiting for commercial release. Waiting for commercial release. But legal battle is going on. Legal battle is going on in Supreme Court. Right. But focus should be here. This is the important one. The last one we can talk about is pest resistance. Crops and pest resistant crops. What is the whole technology? So it was developed by Monsanto, and Monsanto call it BT technology. BT technology. Why they call it BT technology? Because they get the gene from Bacillus thuringiensis. So that's the BT. It's a soil bacteria. And from this soil bacteria, we get a gene that is called CRY gene. What we call it? CRY gene. So CRY gene, different types of CRY genes are there. You don't need to remember the numbers of these genes. It's just like pest resistant crops, Monsanto developed it. Monsanto is USA based company. And BT Technology, because the gene they get to transfer it into the crops are called CRY gene, and this CRY gene is taken from Bacillus thuringiensis. Based on this technology they have developed, the crops they have developed, BT cotton, BT brinjal, and BT maize. I mean, these are the prominent one. This is the only crop, only GM crop accepted in India. So the whole idea is that get the CRY gene, add into the crop. CRY gene will create a CRY protein or BT toxin that will kill the pest. Right. So this is what we have done. This is the most talked thing because obviously this is the only crop we have accepted. So CRY gene will do what? This CRY gene will create a toxin, a protein which is toxic. And this toxic protein will kill pest. So any pests are different. In cotton we have bollworm, in brinjal we have stem and fruit borer. Right. So this is what the pest resistant crops are. So in a nutshell, if we go, then there are few things that we keep in mind. One is BT cotton, the only GM crop India has accepted, and it is being grown larger scale. 96% of the Indian cotton croplands are under BT cotton. The second crop that we should consider is DMH11, because India has developed it, developed in Delhi University, EU, and environmental release has been done. Commercial release is awaited, and if the legal hassle is clear, then we are going to get this as a second GM crop in India. Third GM crop, we should talk about GM rubber, again because of Indian origin, and because it's a non-food crop, the chances are very high that we are going to accept this, because cotton is also non-food GM crop. So non-food GM crop, they don't have any health concerns, right? So the way we have accepted BT cotton, in future we might accept GM rubber. It's like that we have just planted one plant and we are going through the assessment and everything, right? The fourth one we should talk about is golden rice. Golden rice, because India is a very active part of rice genome project that we have conducted long back, and Philippines became the first country. Bangladesh, there the golden rice field trials are going on, right? So probably Bangladesh is going to be the second country to accept it if all goes well, and India is in waiting position. India is also working in association with International Rice Research Institute. We have access to the data of the golden rice, and once we accept this GMH11, then we might go for the golden rice too, right? So from examination point of view, these things are important. Now we'll move on, and we are going to talk about the another area of the important area. Right. So let's talk about gene therapy. Gene therapy. Again, gene therapy is important part. upcoming part. So as a whole, if you look into, right, as a whole, if we look into the entire thing, that gene therapy means you change the genome in order to treat the disease, right? So gene or genome editing for treatment, right? So if a disease is there, disease is linked to a particular gene, then we do genome editing for the treatment purpose, right? And then this gene therapy can be of different types, right? So if we talk about types of gene therapy, then this could be of two types. One is called, the concept is important, and another one is called somatic cell gene therapy. Germ cell gene therapy means that you do some changes in a sperm or ovum, right? So here what we do genome editing in a sperm, ovum or zygote. That means embryo. In basic term, you can call it embryo too. And here what we do, genome editing in body cell. So what could be the impact of it? So this is going to impact the future generation. This is going to impact the present generation only. I'll give you example. It will be clear. So you can write impact on future generation. Here impact on present generation. Present generation only. So present generation only will get impacted. If any kind of benefit they have to get, that will be limited to those only. Disease will progress to the next generation if you go with the somatic cell gene therapy. Right. Example, if we go, then we have TPIVF, or let's say mitochondrial replacement therapy. Very popular thing, 3-parent IVF, 3-parent baby, what we call it. So 3-parent baby is what? It's a germ cell gene therapy, because there you are making changes in embryo, right? Or let's say here the best example that we can have is CAR T-cell therapy. CAR T-cell therapy for the treatment of cancer. So CAR T-cell therapy is for treatment of cancer. This is somatic cell gene therapy. If the therapy goes well, then the person cancer can be eliminated. But if the cancer is having some genetic trait, risk to the next generation will not be eliminated, right? Here what is there, that we are going to change the genetics of embryo, so that particular embryo as a baby will be healthy and all the next generation of that baby will be healthy too. But the problem is, due to safety concerns, accuracy issues, these are largely banned, you can say. So this is controversial, and banned. So controversial and banned, right? And this is allowed. Research is going on, and we are one by one we are basically making it available. ये वाला यूके कर रहा है, नहीं चाइना कर रहे हैं. चाइना भी वो मतलब इट्स नॉट प्रॉपरली बीइंग डन, इट्स अ काइंड ऑफ एक्सपेरिमेंट्स में ही कुछ कर दे रहे हैं. यूके इज द ओनली कंट्री टिल नाउ हु हैज इनेक्टेड अ लॉ टू अलाउ इट, है ना. तो लॉ बनाकर अलाउ किया हुआ है, ठीक है, इन दैट पर्टिकुलर वी कैन डू इट, राइट. So this is a one way to understand gene therapy, right? And TP-IVF is only allowed in UK till now, according to the law. Other countries are contemplating with the idea whether they want to do it or not. CAR T-Cell therapy सब जगह चल रहा है, इंडिया ने भी बना लिया है, नेक्स्ट CR19, है ना. Same therapy we have developed in India also. So this is a one way to understand gene therapy, germ cell gene therapy and somatic cell gene therapy. The another way, another type that we know, that gene is DNA, right? And this will produce what? RNA, and this is going to produce what? Protein, and this protein may cause some damage. Let's say if this gene is faulty, so faulty gene will create faulty RNA, and then it will be creating faulty protein, and this faulty protein will do what? Disease. Because protein is not good, so that will lead to disease, right? So there is one approach that lets stop this particular protein. हम protein बनने ही नहीं देंगे. So in that way, what we can do, if we can remove the gene, right? If we act on the gene, then it is called knock out method of gene therapy. So if you can remove the gene, just you cut the gene, right? There is another method where we don't cut the gene, rather we just destroy the RNA, and that is called knockdown method of gene therapy. So knockout and knockdown, two methods are there. This knockdown method is also known as gene silencing, because you are just silencing the gene. You are doing nothing but silencing the gene. Right. And if we go with the example, then here the best example is CRISPR-Cas9. That's a method that we use. Here the best example is RNA interference. RNA interference. Right. The whole idea is that let's stop this protein production in the body, and it can be done by either destroying the gene by CRISPR-Cas9, cutting the gene out, removal of gene, or by destroying the RNA that is being formed by the DNA itself. Right. So CRISPR-Cas9 is important, and CRISPR-Cas9 is nothing, it's a kind of most accurate scissors to cut the gene out of the genome. Till now, we have not got anything which is as much as accurate than CRISPR or beyond it, right? So most accurate one. We won't go into the details of it because there are, I mean, फिर वो time consuming है, आप पढ़ चुके होंगे. But we can always talk about what are the application of CRISPR. Mainly what happened that whenever we talk about CRISPR, तो हम सिर्फ gene therapy में बात करते हैं. It's not like that. CRISPR is molecular scissors. So it can be used in a variety of ways, not only for disease or gene therapy, but also for GM crops. So we can write just few line. Topic लिखिए CRISPR, Cas9, and you can write this is most accurate. CRISPR-Cas9 most accurate and most advanced. Most accurate and most advanced technology to modify or edit human genome. To modify or edit human genome. Right. Application you can write, just points I'll tell you. Application. And you can write gene therapy. Gene therapy. Second application, cancer treatment. Cancer treatment. Next one, development of GM crops. Development of GM crops. Third one, to get the permanent immunity against viral disease. Permanent immunity against viral disease such as COVID. And one more, to modify microorganism at genetic level. Modify microorganism at genetic level for industrial application. And production of biofuel. Right. So this is one. On the basis of this CRISPR-Cas9 only, there is something called CAR T-cell therapy. CAR T-cell therapy. So what we do in CAR T-cell therapy, we train our T-cells. So T-cells are type of WBCs, and T-cells are genetically modified so that they can recognize the cancer cells in our own body and they can kill it. The whole idea is that T-cell is our own cell. Cancer cell is our own cell. Cancer is nothing but uncontrolled cell division of our own cell. So that is why T-cell, in spite of being the most advanced and most capable WBC, and T-cell can kill anything coming from outside, which is harming the body. But the same T-cell will not be able to kill the cancer cell just because cancer cell and T-cell belongs to the same body. So they have a sense of, self. They consider cancer cell as self only, right? It's a part of our system itself. So what we do some changes in T-cell. We take the T-cell out of the patient's body. We add some genes out there. And then the T-cells are compelled to develop a receptor. And the receptor is called CAR, chimeric antigen receptor, right? So in CAR T-cell, what we do, that this is T-cell, and in this T-cell, let's say this is nucleus. So what we do, we add a DNA, or let's say we add a gene. So this is gene, This is called CAR gene, and this CAR gene is added. We add this. The moment the CAR gene is added, the T cell will convert into this T cell will now convert into CAR T cell. CAR T cell. What is CAR? I'll tell you. CAR is chimeric antigen receptor. Right. Now this CAR T cell, this is, and here you will have the additional segment of the DNA, and because of this it will be having some this is CAR, and this CAR can attach to cancer. So you have given one extra equipment to the T cell. Now this T cell can recognize the cancer cell. Earlier the T cell was having lots many receptor, but not for cancer. Because of one addition of the gene, now this is having receptor for cancer gene, right? So with the help of this receptor, right? So this receptor, it can detect the cancer cell, and T cell will kill the cancer cell. So just like, you need to detect it first. If detection is done, then killing will be done by the T cell. तो किस तरह का है ये? सोमैटिक सेल ही तो है, because you take the T cell out of the patient body, you modify it, and then you put it back. You take few cells, you modify one of them, then in the lab you convert this one cell into millions of the cells, and then you put back into the patient's body. So the T cell is now trained to recognize the cancer cell and they kill it. It's one of the best technology in the recent time that we are using it, and to cut and add these all things, what we do, we use CRISPR-Cas9. Right. So this is CRISPR-Cas9 based CAR T-cell therapy. In 2023, India developed its own CAR T-cell therapy, which are way cheaper than the international one. Right. तो हमने जो डेवलप किया उसका नाम है नेक्स CAR19. नेक्स CAR19. So next CAR19. Next CAR19 is a CAR T-cell therapy. Is a CAR T-cell therapy developed in India by ImmunoAct. ImmunoAct, that's the company's name. ImmunoAct of IIT Bombay. ImmunoAct of IIT Bombay. IIT Bombay based startup. ImmunoAct of IIT Bombay and Tata Memorial Hospital. Tata Memorial Hospital. Right. This is the first indigenous CAR T-cell therapy approved in India. Approved in India. Blood cancer, leukemia. You can write one point. It is being used for the treatment of blood cancer or leukemia. Blood cancer or leukemia. Right. So this is about gene therapy that we should keep in mind. There are few small topics that we are going to discuss, which are very important. One is a stem cell. Again, अच्छा इसमें एक और पॉइंट लिखना. This CAR T-cell therapy. This is also known as cell-based gene therapy. Cell-based gene therapy. Cell-based gene therapy. Cell-based gene therapy. The stem cell are cells in our body which can divide and produce new cells, right? So quickly we can go with what a stem cell can do. What is the importance of a stem cell? Every animal will have a stem cell. And these stem cells are ever dividing cells which can produce new cells into the body, right? And the function of a stem cell, you can write the first one. Function of a stem cell. It supplies new cells. It supplies new cells for embryonic development and growth and development. Embryonic development and growth and development. Growth and development. Right. Second, it supplies new cells for repair and healing. New cells for repair and healing or regeneration, right? Repair, healing को regeneration भी कह सकते हैं. And the third one, it replaces dead cells in the body. It replaces dead cell in the body, right? So this is a cell which will divide and produce different type of cells, and we want to harness this benefit for our own treatment methodology. तो ऐसे सेल्स तो बहुत है, लेकिन दो सेल हैं जिन पे हम बातचीत करते हैं. एक है टोटिपोटेंट स्टेम सेल, another is pluripotent stem cell. So totipotent stem cells are the most potent one. It can produce, it can divide any type of cells in body. But the only controversy it has that we cannot use it because it is available at early embryonic stage. We know that, right? So present only at early embryo. Early embryo. And the only problem with early embryo that if we try to receive and retrieve it, then embryo will be dead. Right. So present only at early embryo, and if we try to retrieve it, we will be damaging the embryo. So we don't use it. It is only for research purpose. Medical application के लिए है, right? So it can produce maximum type of cells in the body, not all, but maximum types of cells in body, right? And retrieval is easy because we can retrieve from umbilical cord blood. Umbilical cord blood. So that is the important one. We know that. So umbilical cord blood is a place where we can get it. That you cannot get that. In spite of being the most promising one, we cannot get that. Pluripotent, the second best, is best in terms of medical application, right? So this dilemma should be very clear in the mind. that the best one we cannot use it because retrieval is not easy and we end up killing and damaging the embryo. So it is prohibited all over the world. But for research purpose we can get it by IVF. लैब में हम फर्टिलाइजेशन करा के ले सकते हैं. So pluripotent stem cell is basically your main cell. This is the important one. All over the world for medical application, the best you can use is pluripotent, right? But then you get the only one chance to get the pluripotent stem cell, that is at the time of birth when you are going to cut the umbilical cord. Easiest way यही है. बहुत एक्सटेंसिव रिसर्च में आप निकाल सकते हो pluripotent stem cells बर्थ के बाद भी, but it is going to be very tough task. The cost wise it won't be feasible one, and then you are not sure that you are going to get it or not, है ना. तो इसका एक और तरीका डेवलप हुआ, नोबेल प्राइज मिला उस पे, and that is called induced pluripotency. Pluripotency. Induced pluripotency. And same thing is also known as or induced pluripotent stem cell, or it is also known as IPS cell. IPS cell. So what we do, a normal cell can be converted into pluripotent stem cells by the help of gene reprogramming. So you can write a normal cell of the body, can be converted into pluripotent stem cell. Pluripotent stem cell by the process of gene reprogramming. For this achievement, Nobel Prize of Medicine was given. Nobel Prize of Medicine was given to Japanese scientist Shinya Yamanaka in 2012. So that is one thing you should be knowing, right? One more area that I just want, obviously the applications, we know that applications could be that we can treat lot many disease related to body and tissues. डायबिटीज को रिवर्स कर सकते हैं, इन फ्यूचर टिश्यू डैमेज को ठीक कर सकते हैं, राइट. इन फ्यूचर वी कैन डेवलप ऑर्गन्स आउट ऑफ इट. So that's entire list. But then there is another one that is called cloning. I won't go in detail of it, but you just need to know the dimension of it. So remember, this cloning can be of two types. One is called therapeutic cloning. People have tendency to forget it because we always focus on this part of the cloning that is called reproductive cloning. Right. So therapeutic cloning is equally important, and there we can do gene cloning, we can do tissue cloning, we can do cell, tissue and organ cloning. Right. Gene cloning, the example should be there. and insulin production. So gene from one organism will put into the another cell, and then this cell will be divided. For that matter, if you look very carefully, then even your CAR T-cell therapy is going through gene cloning only, because you have made one cell, and then this one T cell in the lab multiplied into millions of T cells. So you added one gene, one CAR gene, but when the T cell is multiplying, then what is happening? Multiple copies of this, CAR gene is happening, right? So you will have to, जितना पढ़ा है उतना ही नहीं, रिलेट भी करना पड़ सकता है, कुछ नया भी परम्यूटेशन कॉम्बिनेशन वो दे सकते हैं. तो इंसुलिन इज अ क्लासिक एग्जांपल, बट इन सम सेंस CAR T cell therapy, there also we are basically making genes clone in the T cell, because T cell to hai, we have done with the T cell that we have added some gene into it, and once the T cell will divide, the gene will also divide. So multiple copy of those genes and the T cells has been developed. This cell tissue and organ cloning is nothing but the stem cell therapy. And reproductive cloning is again, that is organism cloning. And India has done a lot into it. So we won't go into the details of it, but we have done animal cloning, buffalo, cow, Pashmina wool, goat, right? So we have done many of them. And the recent was this Ganga Gir cow clone that we have cloned few years back only. So this is something in a cloning that you should be doing. Focus on this area. Ye bhulna nahi. And maybe, when CAR T-cell therapy and all, they can link it with the clone. So just when you get something like that, you need to take a pause to link the things. Reasoning develop करनी है. Anyway, तो अभी आगे मूव करते हैं. Next topic that we are going to discuss, next chapter, is again nanotech. So nanotechnology. Basic concept we know that it remains same, that at 10 to power minus 9, that is nanoscale. 10 to power minus 9 meter we can say, that is called nanoscale. What will happen? Property of material will change. So property of material will change. And this changed property can be utilized to develop different kind of systems and process, that is called nanotechnology. Right. So in nanotech, because it's emerging technology, they can ask question from the very basic thing, right? Because obviously it's a new technology, so very basic things can be asked, and many times they have asked application based question also, like fuel cell वगैरह पूछ चुके हैं पहले भी, right? So that's the one thing. Second thing that we should be knowing, that how to bring down something to the nanoscale, right? Nanoscale means 10 to power minus 9. In that term, we can call it like, when we say nanoscale, so anything below 100 nanometer. So below 100 nanometer is your nanoscale, or let's say we call it like 1 nanometer to 100 nanometer. So even if we say 1 nanometer to 100 nanometer, it is below 100 nanometer. So the moment you go beyond 100 nanometer, you are in micro scale. 100 nanometer and down, you are at nanoscale. And at nanoscale, the things will change because of the lots of things happening at the surface level, surface and volume ratio, quantum mechanical phenomenon, all things will start coming in, and then it will be there. Now, how to reach to the nanoscale? How to reach to nanoscale? We have talked about these two. There are two methods. One is top-down method. Another is bottom-up method. Bottom-up method. So top-down method is miniaturization. That we call it miniaturization. That means you keep on decreasing size. So top-down is that you keep on decreasing size. You cutting down the things till the time you reach to that particular way. Bottom-up is you start from atom. So you start from atom and then you reach to the nanoscale. Here what you are going? You start from bulk and then you reach to the nanoscale. So this is big 2 nanoscale and this is atom atom 2 nanoscale. Future lies over here because this is the important one. मैंने एक पॉइंट आपको बताई थी कि टॉप डाउन is not useful for mass production. Mass production means one go you want to produce many things. Whereas bottom up is good for mass production. It's a rapid and mass production because nano systems are so small that 1 or 2 nano systems will not be working for us. If we want to use nanomedicine in our body, millions of nanomedicines molecule would be needed in one dose, right? And now this can be done only with the help of bottom up, right? So you can write एक point लिख लीजिए नीचे इसी के bottom up is futuristic and more promising. Bottom up is futuristic and more promising. Because it is based on molecular assembly and chemical synthesis. Molecular assembly and chemical synthesis. And this is suitable for mass production. सिंपल टॉप डाउन ठीक है, टॉप डाउन भी हम यूज करते हैं. हाँ कर सकते हैं हम, कोई इशू नहीं है. लेकिन प्रॉब्लम है कि सिंपल स्ट्रक्चर से हम क्या अचीव करेंगे, वो तो हम कर चुके हैं. तो नाउ हाँ क्या हाँ यदि कॉम्प्लेक्स के लिए होगा तो मास अभी आपने कंप्लीट नहीं किया ना. मास प्रोडक्शन ऑफ कॉम्प्लेक्स स्ट्रक्चर्स. मास प्रोडक्शन ऑफ कॉम्प्लेक्स स्ट्रक्चर्स. तो कॉम्प्लेक्स स्ट्रक्चर ही फ्यूचर है. नैनो मशीन्स एंड नैनो सिस्टम्स आर द फ्यूचर. सिंपल तो बना ही चुके हम बहुत सारे, है ना. तो दैट इज वन थिंग यू शुड बी नोइंग. द सेकेंड थिंग जो बहुत इजी है बट इंपॉर्टेंट है, आपको फोकस रखना है कार्बन नैनो स्ट्रक्चर्स पे और नैनो मटेरियल्स पे, राइट. बिकॉज पीटी ओरिएंटेड ही इंफॉर्मेशन है इसमें. एंड देयर आर 3 थिंग्स वी नो. ग्राफीन, second one is TNT, third one is fullerene, fourth one is dendrimer, जो बेसिकली आपको पता है. Nowadays the dendrimer तक ही रखो, बाकी हम आगे देखते हैं. तो one thing is common, that these all are purest form of carbon, है ना. Dendrimer क्या है? It's a branched structure of CNT. So branched structure of CNT is dendrimer, and this is going to be 3D structure. This could be big enough to be, visible to the naked eye. क्योंकि वो तो ब्रांच स्ट्रक्चर है ना, बहुत सारे CNT को मिला दिया, जैसे कि बहुत सारे एटम से मिलके ही कुछ भी बना हुआ है. तो इंडिविजुअल एटम वी कैनॉट सी, but the anything which has been developed by or made by assemblage of atoms we can see. Bricks और units कितने छोटे हैं उससे फर्क नहीं पड़ता. If you make a complex structure out of it, you will be able to see that. ग्राफीन के बारे में मैंने कहा था कि this is 2D and it is made up of carbon hexagon, if you remember. So this particular information should be clear. One carbon attached to 3 carbon electron, 3 with them. CNT is 1D structure, that's a rolled up graphene. So this is rolled up graphene. It's a pipe like structure made of graphene. Fullerene is 0D structure, and fullerene, one thing is there in fullerene, here I am writing. किसमें देखो आप, ये दोनों किससे बने? Made up of carbon hexagon. This is carbon hexagon, right? So that is structure. You have to when you look into the copy, you can find it, right? So that's about carbon nanomaterial. Now, the most important application of it, if you go with the application, then the application, जो मेजर एप्लीकेशन है उसको एक जगह लिस्ट करते हैं, है ना. जैसे एक एप्लीकेशन है हेल्थ का. Two of them are very important. One is called highly sensitive disease detector, and the second one is targeted drug delivery method. Right, these both will create precision medicine. Right. So be open for new terminologies the UPSC can throw. Try to understand the terminology, that what exactly it means. Then you look into your knowledge bank, that have we read something in that line, and then you can see that it is reflecting in the option also. So options are very important guide, है ना. कई बार क्वेश्चन पढ़ के नहीं लगता कि होगा, फिर ऑप्शन पढ़ के लगने लगता है कि ये हो जाएगा. एंड इट्स इन अ वे इट्स इजी. आप ये सोचो पीटी, obviously, I mean nowadays we'll say that PT is the most difficult part of this examination, but मतलब अगर आपकी तैयारी अच्छी है तो डिफिकल्ट इसलिए कि कट ऑफ बहुत अच्छा नहीं जाता. आई मीन कट ऑफ इज टू लो, तो आप जितना बनाते नहीं उससे ज्यादा नहीं बनाते हो, तो डिफिकल्ट. मेंस में तो है ना कि क्वेश्चन तो लिख के ही आते हैं हम, बट द क्वेश्चन दैट यू कैन क्रैक, वो देखो उसमें क्वेश्चन भी है और आंसर्स भी है, रिलेट करना है बस. तो it's like we always say that in prelims, two things matter. The first thing, that what you have studied till that date, जिस दिन प्रीलिम्स देने जा रहे हो उस दिन तक क्या क्या कितना पढ़ा है. और सेकंड थिंग मैटर करता है कि जिस दिन प्रीलिम्स देने जा रहे हो वो दिन कैसा है ना, वो भी मैटर करता है. कि उस दिन, वो दिन का मतलब लक की बात नहीं कर रहा, उस दिन आप कैसे हो, व्हाट इज योर मेंटल स्टेटस, राइट, क्योंकि वो बहुत डिसाइसिव हो जाता है. समटाइम पीपल आर इन स्ट्रेस, कुछ भी ऐसा इस तरह से, वो इट विल बी इम्पैक्टिंग द डिसीजन मेकिंग बिकॉज यू आर अंडर प्रेशर, है ना. वो 2 घंटे का टाइम, उसी में सब कुछ करना. तो दैट इज वाई से अभी जो भी लोग अभी मिल रहे हैं ना, मैं सबको कह रहा हूं कि अब पुराना पढ़ने का कोई मतलब नहीं है, मतलब नया पढ़ने का कोई मतलब नहीं है. All you need to do that you have to revise, and one thing you can read that is current developments जो आपने बना रखा हुआ है, आपके पास compilation है, you have it, right. And even I mean people who are in the race, they have already done current affairs once, so the only thing now you can do is to revise, है ना. ऐसा लगता है कि now this is something that I should read, ये भी करेंगे, वो लास्ट डे तक लगता रहेगा, but it's never ending, right. Anyway, so health and medicine, highly sensitive disease detector, targeted drug delivery method. Examples we have talked about, not necessarily that they are going for the example, rather they go for the concept. है ना? Like fuel cell technology, they have asked question, they have gone to the concepts, not for the examples and all. Examples तो आपने लिए रखा मेंस के हिसाब से. If you want to write something in mains, then obviously examples would be handy. But here everything is related to what? कॉन्सेप्ट, हाईली सेंसिटिव डिजिटेटर. अब देखो ध्यान से, तो कार टी सेल थेरेपी में हमने जीन्स डाले थे, जीन्स से उसके ऊपर क्या हुआ था, एक सेंसर बना था, सेंसर जिसका नाम था CAR, काइमेरिक एंटीजन रिसेप्टर, मेड अप ऑफ प्रोटीन, राइट. इफ सिमिलर थिंग वी डू विद द हेल्प ऑफ नैनो फैब्रिकेशन, देन इट इज नैनो टेक्नोलॉजी. यहां पे भी सेंसर ही बनाना है, रिसेप्टर ही बनाना है, बट देयर रिसेप्टर वाज मेड बाय DNA to RNA to protein, that method, right? That is central dogma of the biology. DNA induced the formation of receptor. So we call it CAR T-cell therapy. The same thing we are doing here in nanotech, but in a direct way. And when we do it in a direct way by nano fabrication, we are coming down to the nanoscale because cells are also functioning at nanoscale. And then we are manipulating the cells or the sensors in the body, and it is called nanotechnology. इसलिए मैंने कहा था कि देयर इज समथिंग कॉल्ड बायोटेक, देयर इज समथिंग कॉल्ड नैनो बायोटेक. बायोटेक इज एंजाइमेटिक मेथड, बायोटेक में हमेशा जीन्स और एंजाइम की बात होती है. नैनोटेक में डायरेक्ट मेथड है वो. देयर वी आल्सो मैनिपुलेट विद द सेल्स ओनली, सेल सिस्टम्स ओनली, बट वी मैनिपुलेट डायरेक्टली विद द हेल्प ऑफ नैनो स्ट्रक्चर्स. हम वहां पे नैनो स्ट्रक्चर्स को मैनिपुलेट करते हैं, है ना. आई डोंट थिंक दैट आई नीड टू गो इन डिटेल विद इट, जिसमें हमने डिस्कस किया था. एक सेंसर होगा एंड दिस सेंसर विल बी एबल टू catch the disease, because if a cancerous cell is there, then cancerous cell will be having a particular protein structure or surface protein or antigen on its body, and this will go and attach to it. And if we add a drug carrier molecule with this, तो ये बन जाएगा targeted drug delivery system. So now it will have sensor plus drug carrier molecule put together. It has turned into targeted drug delivery method. Second area that we should be aware of is environment and energy. We can keep it together. And the prominent technology over there is nano solar cell, and then there is fuel cell. So nano solar cell and fuel cell. Fuel cell they have asked. Nano solar cell they might ask now. I mean, you never know. So graphene based or CNT based solar cell which can harvest energy from both visible light and infrared. Energy potential will be double, surface area will be higher, conductivity will be so well that resistance will be very low, overall output would be so much. That is why nowadays we are looking into the news and all. तो वो कहते हैं कि हम सेल्फ सस्टेन बिल्डिंग बनाएंगे एनर्जी के लेवल पे, है ना. तो बिल्डिंग्स विल बी सेल्फ सस्टेन इन टर्म्स ऑफ एनर्जी, एंड देयर फोकस फॉर दिस इज सोलर एनर्जी, है ना. अब तो ऐसे सीमेंट बना रहे हैं, तो सीमेंट इलेक्ट्रिसिटी जनरेट करेगा, राइट. सो इफ यू गो इनटू द कोर ऑफ दिस सीमेंट एंड ऑल, इट्स अ मटेरियल साइंस एट द एंड ऑफ द डे, and this material science is linked with what? Nanostructures, right? So that is one area, that energy part. And the another area that we can add over here is environmental protection. And here we use nanochemical sensor for removal of chemical pollutants. Chemical pollutants. Removal of chemical pollutants. Right. And that is what we call it, environmental cleanup. Environmental cleanup by nanosensors. So we can clean something out of the environment. Right. एक एरिया इस पे थोड़ी बहुत कुछ आया करंट में, और वो है सेफ ड्रिंकिंग वाटर बाय नैनो फिल्टर्स. तो नैनो फिल्टर्स, right? So nano filters are being considered as good option for the other filtration mechanism that we are having, because nano filters, एक तो हम इसको ऐसा भी बना सकते हैं कि बिना एनर्जी के चले, पैसिव फिल्टरिंग करे, है ना, ग्रेविटी पे. इट्स लाइक अ नॉर्मल फिल्टर, यू पुट द वाटर, वाटर विल ड्रिप डाउन. सेकेंड थिंग, नैनो फिल्टर्स कैन टेक केयर ऑफ बोथ biological contamination and heavy metal contamination, right? ठीक है, अगर आप चाहते हो कि इसकी टेस्ट भी डेवलप हो जाए बढ़िया, तो फिर तो आरओ का सिस्टम लगाना पड़ेगा with the nano filter itself. Even the filter into the RO system, yeah, right? But that not advanced nanotechnology. It's like mainly filtering on the basis of reverse osmosis to eliminate all the salts to improve the taste of the water. लेकिन अगर हम पैसिव फिल्टरिंग में जाएं तो एनर्जी इज नॉट रिक्वायर्ड. यू जस्ट अलाउड वाटर दिस टू पास थ्रू द नैनो फिल्टर्स, एंड नैनो फिल्टर्स विल टेक केयर ऑफ बायोलॉजिकल कंटामिनेशन एंड द हेवी मेटल कंटामिनेशन, एंड दैट इज इंपॉर्टेंट. हेवी मेटल को बचाना इंपॉर्टेंट है. इंडिया में इस पे भी पीटी में क्वेश्चन था कि पंजाब स्टेट के नाम से पूछा था कि यहाँ के पानी में क्या प्रॉब्लम है. यूरेनियम, आई मीन देयर इज अ पार्ट्स ऑफ पंजाब वाटर वेयर यूरेनियम इज देयर. And uranium, cadmium, arsenic, lead, these all are heavy metals, and these metals can do a lot of damage into the body. किडनी विडनी तो कॉमन डैमेज है, कैंसर वैंसर सीधा करता है ना. तो दैट कैन बी टेकन केयर ऑफ. द अनदर एरिया दैट इज इंपॉर्टेंट इन नैनोटेक, जो मुझे लगता है कि इंडिया एग्रीकल्चरल कंट्री है, तो नैनो एग्रीकल्चर. And why it is important, you know it, because हमने दो फर्टिलाइजर्स बना दिए नैनो वाले. एक फर्टिलाइजर जो है वो हमारा है नैनो यूरिया लिक्विड, एंड द सेकंड वन इज नैनो डीएपी, है ना. So if you remember, we have divided into three parts. One is crop improvement. Crop improvement. The second area is crop protection, and the third area is crop growth and development, or productivity, जो भी आप कह सकते हैं. Growth and development is okay, where This is Nanobiotech. Nanobiotech. Crop protection is nanopesticides. And this is nanofertilizer. And there we are having two examples, nano urea liquid and nano DAP, diammonium phosphate liquid. Both has been developed by IFCO. Right. These all are in experimental state. Although many countries are engaged into it, they are developing new crop variety by using nanotechnology. Like I told you that if you add genes by enzymatic methods by using enzyme like ligase, restriction endonuclease, or you use CRISPR-Cas9, there also enzyme is there. Cas9 is enzyme only, right? तो बायोटेक है, लेकिन डायरेक्टली अगर आप पुश करते हो by using the nano holes, nano instrument, reaching to the direct gene level, then it is nanobiotech, and we are able to manipulate that. अब हम कर पा रहे, right? Nanopesticide is again targeted delivery system. The whole idea is that pesticide would be targeted to only those pests that we want to kill, those insects which are pests basically. So like we have targeted drug delivery system, we'll have this. अब क्या है ना, इस पूरे प्रोसेस को आप डाल सकते हो कि इसमें प्रिसीजन एग्रीकल्चर में भी. I mean, precision agriculture is not only robotics. कहीं भी प्रिसीजन है तो वो एक बड़ा ब्रैकेट बनता है. So nowadays two things are very important. One is precision medicine. अब क्या है कि प्रिसीजन मेडिसिन अगर आ गया क्वेश्चन में, तो क्वेश्चन डिसाइड नहीं करेगा कि आपसे बनेगा कि नहीं बनेगा. ऑप्शन विल डिसाइड whether you are able to relate with that, and it won't be that one question and one answer. ऐसा नहीं होगा. One question and multiple answers you have to. अब तो और भी खतरनाक करते हैं ना वो. पहले तो था कि चलो 1 and 2, 1 and 2 and 3, अब तो बोलते हैं any 2, any 1, any 3. So that, I mean, obviously they will keep on raising the bar. अब क्या है ना, कई बार वो फेयर तरीके से बार को रेज करते हैं. अब जो कर रहे हैं वो थोड़ा अनफेयर टाइप से है. अनफेयर का मतलब कि दे आर मेकिंग इट डिफिकल्ट, दे आर मेकिंग इट टू द लेवल वेयर कंफ्यूजन विल गो हाई. तो अनफेयर तो कुछ भी नहीं है. इट्स यूपीएससी, दे कैन डू एनीथिंग, बट देन कंफ्यूजन लेवल बढ़ा देंगे. तो उनको लग गया कि एलिमिनेशन से ये लोग निकाल दे रहे हैं, है कि नहीं. तो दे एलिमिनेटेड द एलिमिनेशन मेथड इटसेल्फ, कि चलो खत्म ही करते हैं. अब कोई लिख रहा है एनी 2, अब एलिमिनेशन कैसे करोगे. नाउ यू नीड टू बी एब्सोल्युटली श्योर अबाउट कि ये वाला स्टेटमेंट तो नहीं होगा और ये दोनों होगा, राइट. तो कंफ्यूजन लेवल विल बी हायर, चांसेस ऑफ गेटिंग रॉन्ग विल बी हायर. लेकिन इससे क्या होगा, फिर वही है कि वी प्लान, वी स्टडी, वी प्रिपेयर आवरसेल्फ, वी एक्सपेक्ट दैट कट ऑफ विल गो अप, बट ओवरऑल कट ऑफ वही रह जाता है कि नहीं. क्योंकि हम भी तैयार करते हैं हर, I mean, every next year, your preparation level, as a teacher, I can feel that with every passing year, the level of preparation goes up. And nowadays, it's going up by every 6 months because of the information flow. अब तो मैं तो बड़ा हैप्पी रहता हूं इसलिए कि अब तो गूगल पे सर्च करो तो एआई उसका रिप्लाई कर रहा है. एक दो बार तो मैंने ऐसे कहा और यूपीएससी लगा के सर्च किया, वो पूरा उसी फॉर्मेट पे दे दिया. Introduction, application, issues, right? Obviously, if information is now easily accessible to you, तो फिर वो डिफिकल्टी लेवल किसी और कॉन्सेप्ट पे बनाएंगे, राइट. तो इट्स ओके, कोई बात नहीं. कट ऑफ is not going to improve because of the difficulty level and preparation level, both are going side by side. वो साथ ही साथ चल रहे हैं, राइट. अंडरस्टैंडिंग क्लियर होना चाहिए. सो दिस nano urea liquid, nano DAP liquid, both was developed in India first of its own kind in the entire world. IFCO एक नाम याद रखना, IFCO has developed it, Indian Farmers Fertilizer Corporation, and benefits, you know that because they are very handy, easy to transport. 500 ml का bottle आता है, cost effective. It's also impacting on, positively impacting on the import of urea and fertilizers that we do, वो भी कम हो गया है ना. Although nowadays there is a new problem that farmers are complaining that it's not functioning or not working in the way that has been promised. इफको का कहना है कि ये काम तो ठीक करता है, अभी farmers are not used to of it, right? Once they get used to of it, they will be able to get best out of it. क्योंकि उन्होंने जो किया उसमें तो ठीक था, उन्होंने कहा कि 8% ओवरऑल प्रोडक्टिविटी बढ़ रही है सिर्फ इसके कारण. So this agriculture part you should be knowing. The another part that we can talk about is textile. Why textile? Because their Indian company has done some wonderful job. वो एक कंपनी है एडवांस इंटरनेशनल रिसर्च सेंटर फॉर पाउडर मेटलर्जी एंड न्यू मटेरियल. तो एडवांस इंटरनेशनल रिसर्च सेंटर फॉर पाउडर मेटलर्जी and new material, Telangana. And the important thing is that this organization, they also work in association with DRDO, है ना, उनके लिए भी बहुत सारे मटेरियल बनाती है. Because material science is very important and DRDO need to have access to the best of the material for the arms and ammunitions also. So that one thing you should be knowing, you can write it. Okay, this organization works in association with DRDO. DRDO. And they have developed different types of textiles, and fabrics, based on nanomaterials and nanotechnology. Nanomaterial and nanotechnology. Right. For example, you can write antimicrobial fabric with silver nanocrystal. Antiviral mask with copper oxide nanocrystal. Right. Next, technical fabric for armed forces and firefighters. Technical fabric for armed forces and firefighters. Right. One more you can write sun wash fabric or self cleaning fabric. sun wash fabric or self cleaning fabric? Right. So that we have done. Now in nanotechnology, the development part should also be important. So development of nanotechnology in India. Simple है but important है. One is NSTI, Nanoscience and Technology Initiative. We have done it, right? And the second one is NSTM. We are going through it. Nanoscience and Technology Mission, and we call it Nano Mission. So we are going through this Nano Mission. Objectives should be very clear that Nano Mission has got objectives to promote this nanotechnology in 5 different areas. One is fundamental research को boost करना है, infrastructure development and development of ecosystem, development of trained human capital or human resource, very important, inclusion of private sector, right? Anything, even in robotics also we are seeing that inclusion of private sector is being promoted, and international collaborations, right? ये सब हम कर रहे हैं. For international collaboration, इसमें एक और aspect है, international collaboration in nanotech. So there we are having IconSat, if you remember, International Conference on Nanoscience and Technology, IconSat. Right. Then few information was there that Nano Park is being developed in Bangalore, that we know. Indian Institute of Nanoscience and Technology, INST, that was established in Mohali, the first one, and we are developing 3 more now, right? फिर एक नैनो इलेक्ट्रॉनिक सेंटर डेवलप किया IIT बॉम्बे, IIT दिल्ली एंड IISc बैंगलोर में. तो नैनो इलेक्ट्रॉनिक सेंटर promote nanotechnology in electronics and all, right? तो ये पॉइंट लिख लेना आप. नैनो इलेक्ट्रॉनिक सेंटर, nano electronic centers have been established at IIT Bombay, IIT Delhi and IISc Bangalore. IIT Bombay, IIT Delhi and IISc Bangalore to promote nanotechnology in electronics. By using graphene, CNT and other nanomaterials. Graphene, CNT and other nanomaterials. Right. Robotics is one such area जहां पे पीटी में बहुत सवाल नहीं बनते, है ना? बट फिर भी एक बार आप पढ़ना उसको, just go through once, especially the application part, right? Any new robot which came into the news, the type of robot in the, I mean, example wise, so it's very difficult because there are hundreds of robots which are coming, right? But yes, industrial application of robot is one thing that you should be aware of. We also talked about this World Robotics Report that is important one, you should be aware of. But robotics का track record PT में ऐसा है कि they have asked no question from robotics, है ना, कुछ पूछा नहीं, मेंस में ही उन्होंने पूछा. So we skip robotics part and we'll move on to the space tech. Right. डॉग वाला ना, हाँ, नाम तो नहीं पता बट वो कैमरा लगा हुआ है, डॉग लाइक रोबोट 4 फुटेड, है ना. अच्छा, okay. So in a space tech there are two areas. The first area, I mean one thing that we should be aware of, एक तो पहला outer space treaty, and why it is important I'll tell you. It was in 1967, right? So long back we signed it under UN, but now this has become important once again. This has been important all the while because out of this we came up with Artemis Accord. Artemis Accords. And Artemis Accords came only in 2020 and India became part of it in 2023. So that is why this entire thing is important. Artemis Accord is strategically important for India. I mean, first thing that India is always committed for peaceful application of space technology. So India is always committed for peaceful application of space technology, that is there. But like other country of the world, they have developed many space technology which are not so peaceful. India has also developed. तो फिर वो dilution जो हुआ सबके लिए हुआ, इंडिया के लिए भी हुआ. And then USA came up with this Artemis Accord which are non-binding one. UPSC में जब मल्टीपल क्वेरी क्वेश्चन बनाते हैं ना तो ये भी पूछना शुरू कर देते हैं, बाइंडिंग है कि नॉन बाइंडिंग है. इन्वायरमेंट में पूछते हैं. So if some accords and some conventions are there, then they can ask, they can put just one, true and false में. So non-binding Artemis Accord, it's a kind of, you know, on your wish you want to come in or not, right? Moreover, India at the very strategic point entered into Artemis Accord. क्योंकि Artemis Accord में एक आपने देखा होगा कि there is one provision that is of data sharing, right, and development of common infrastructure in a space. So data sharing means that if a country has done some mission, then that data would be shared with the member country, because now we are re-establishing a space into the peaceful way. तो उसमें वो unnecessary competition जो होता है उसको eliminate करो. Let's put the collective effort. India is now planning to go for the Gaganyaan, और गगनयान इंडिया का ह्यूमन मिशन होगा. Rather, इंडिया अपनी फुल कैपेसिटी में जितना कुछ कर सकता था कर चुका है कि नहीं. चंद्रयान 1 we have done it. We did, I mean we send our spacecraft to moon orbit. Mangalyaan 1 we have done it. उससे भी जो important था landing on the moon, that we have done it. Now anything you do beyond require some more expertise. If you get the data, you get the, you know, technology from the other country, it would be easier for you. अभी जो next lined up है उसमें एक जैसे वीनस ऑर्बिटर मिशन है, ठीक है, वो हम कर लेंगे क्योंकि हम ऑलरेडी MOM कर चुके हैं, हम चंद्रयान कर चुके हैं. But when it's come to landing onto the Mars, it's going to be difficult one. In moon also the landing was not done in a one, एक बार में नहीं कर पाए. And the most important one that India is now planning to go for Gaganyaan, already कर चुके हैं. So when Gaganyaan is going, when 3 human beings are being sent, then we cannot take any chance. उसमें ये future prospects अगर देखें तो that is very good. Strategically, India decided in 2023 that India will become Artemis Accord member, right? and will help this entire re-establishment of the foundation of outer space treaty. वो हम करने की कोशिश करेंगे. So that is one thing you should be aware of. और commitments क्या है Artemis Accord में, that's very important. So the commitments are very clear, like I told you that in Artemis Accord there are commitment first, that peaceful application of space in accordance with international law. And law is what? Outer Space Treaty. Second is development of common infrastructure so that we can enhance the scientific discovery and commercial utilization. Third one is data sharing with member countries that will help those countries to explore space in a better way. Then very important concept is preservation of heritage sites, है ना. तो नए terms जब आते हैं, they have potential, because new terms have potential to be asked maybe this year or next year, जब भी. So preservation of heritage site in a space, like landing sites, artifacts, and the objects that we have placed out there, which are basically evidences of human activities there. Utilization of space resources, optimal utilization without interfering with other countries' missions and activities. And the last one is mitigation of debris, very important. उसमें भी हम इसको यूज कर रहे हैं, right? So that's one thing you should be aware of. Now, if we go with the other next important topic, तो जो next important topic है space tech का, उसमें launch vehicle is important, right? तो launch vehicle का एक track record है पूछने, they ask, right? And we have divided this launch vehicle into different way. जैसे कि हमने divide किया था sounding rocket, right? Not important. Sounding rocket, you should be knowing that sounding rocket is one such rocket which can be only be used for atmospheric studies, not for satellite launches. But India started its journey in rocketry with the sounding rocket only. When we come to the launch vehicles, then sounding rocket is not part of launch vehicle. तो launch vehicle में past launch vehicle कौन से हमारे? SLV and ASLV. तो ये भी नहीं पढ़ना है, बस जो भी पढ़ा है काफी है. If we talk about present launch vehicle, then it is PSLV, GSLV Mark II, then LVM3, SSLV, that is also there. हाँ, ये आपके present launch vehicle है. And in future if we go, so we have already experimenting with RLV, और अभी NGLV हमने डेवलप करना है, जिसको सूर्या रॉकेट भी कहते हैं. तो NGLV और सूर्या, right? So now these two is important. LVM3 is important, और LVM3 का एक वर्जन डेवलप किया है, दैट इज कॉल्ड HRLV, ह्यूमन रेटेड लॉन्च व्हीकल. And this HRLV will be used in Gaganyaan. So this is being used for Gaganyaan mission. So the rocket systems which are important is RLV, NGLV, SSLV और LVM3, because this RLV is a part of it. मतलब गेस वर्क नहीं है, कोई भरोसा नहीं है. समटाइम यूपीएससी, अब NGLV बहुत न्यूज में है, तो एक टेंडेंसी ये भी होती है कि जो बहुत प्रोमिनेंटली न्यूज में मत पूछो, रेदर गेट इनटू द पास्ट ऑफ द एंटायर थिंग, और कुछ ऐसे क्वेश्चन बना देंगे जो पास्ट और रॉकेट की इवोल्यूशन पे बेस्ड होगा. यू गेट माय पॉइंट? So in that way, what will happen? This even SLV, ASLV will be important, because they will track the evolution, how the rocket has evolved in India right? In that evolution, 4 statements are there, and they have placed 1 or 2 statement in a way that वो वहां पे आपके काम आएगा. जैसे कि मैं एक सिंपल एग्जांपल देता हूं. इनिशियल रॉकेट्स में सॉलिड फ्यूल इस्तेमाल हुआ था, तो सॉलिड फ्यूल, एंड देन आफ्टर सॉलिड फ्यूल we started using liquid fuel. You get my point? In PSLV and then afterwards. So this is evolution of rocket, right? And if they put one statement, then in the development of launch vehicles, India started with solid fuel and gradually evolved to liquid and cryogenic fuel. ठीक है ना? सही है. But for that NGL, sorry, SLV, ASLV important हो ही गया. And this is the one statement that I have made over here. उसी में से वो कुछ डाल देंगे कि ठीक है, SLV, ASLV आर बेस्ड ऑन लिक्विड फ्यूल. ट्रू एंड फॉल्स, तो फॉल्स करना पड़ेगा, राइट? सो दे आर नॉट गोइंग टू आस्क पेलोड कैपेसिटी एंड ऑल in terms of the past launch vehicles, because that's nothing. It's a, but yes, fuel is very important concept of the rocket system. Everything is moving around the fuel only, right? If you go into the history, we had a lot of problem while developing GSLV and LVM3 because of the cryogenic, and PSLV was based on the liquid fuel Vikas engine. So that is important one that you should be knowing. So as a whole, if we go, then GSLV, sorry, LVM3, that is one. The second one is SSLV. Third one is RLV. And the fourth one is NGLV. NGLV is particularly more important because NGLV is going to use semi-cryogenic engine, expandable launch vehicle which can carry up to 10 tons of the payload and 30 tons into the LEO. और इन फ्यूचर हम इसको बना लेते हैं तो ये सबको हम रिप्लेस करेंगे. सो यू कैन राइट फ्यू पॉइंट्स. NGLV. NGLV is being developed under Project Surya. NGLV is being developed under Project Surya. Second point. This will be 3-stage launch vehicle. 3-stage launch vehicle. और एक टर्म यूज करते हैं 3 stage to the orbit. In bracket you write 3 stage to the orbit. So that is a term they use, 3 stage to the orbit. Next you can write in future it can replace PSLV, GSLV and LVM3. Next point. NGLV will be expandable launch vehicle, which can carry which can carry 30 tons payload to LEO, 30 tons payload to LEO, and 10 tons payload, 10 tons payload to GSO, sorry, GTO. एक मेजर चेंज जो हम कर रहे हैं इसमें, दैट फर्स्ट स्टेज सॉलिड जो होता था उसको रिप्लेस करके सेमी क्रायोजेनिक इंजन. And this will have impact onto the entire system. Whether it will also help us to mitigate with the pollution that occur during the launch of the, vehicles. So you can write NGLV will have semi-cryogenic engine in first stage with liquid kerosene or methane, with liquid kerosene or methane as fuel and liquid oxygen as oxidizer. Liquid oxygen as oxidizer. Full stop. This will minimize the pollution, which occurs during solid fuel first stage. Solid fuel first stage. Right. Next point you can write. First stage will be reusable stage. Reusable stage. And it will minimize the cost. It will minimize the cost. Right. One more point. This launch vehicle will play important role in development of भारतीय अंतरिक्ष स्टेशन, by 2035 and human lunar mission. Human lunar mission by 2040. हाँ, चंद्रयान 5 3 हमने भेज दिया, 4 होगा सैंपल रिटर्न मिशन और फिर जो लैंडिंग मिशन होगी ह्यूमन की वो होगा चंद्रयान 5. राइट. So this is about our NGLV, the major points that we have done. RLV we have done it, है ना, because I'm assuming that जो भी क्लास ले रहे हैं कर रहे हैं उनके पास नोट्स है ना, और अगर मान लो 5% लोग जिनके पास नहीं है आप नोट्स ले लेना, otherwise it will make no sense, right? So we won't go into that. Yes, one SSLV is again very important because of India's capability to launch one launch per week, है ना. तो SSLV की significance इसमें है कि that has opened the door to the private sector to enter into the ISRO to develop the launch vehicles at mass scale. तो एक पॉइंट SSLV के लिए लिख लो so that you can keep in mind. SSLV 3-stage launch vehicle. 3-stage launch vehicle. All solid. 3-stage launch vehicle. All solid fuel. Right? Next point, it can be assembled in 72 hours. It can be assembled in 72 hours. Next, ISRO has achieved one launch per week capacity. One launch per week capacity with SSLV. And one more point, private companies are being engaged. Private companies are being engaged for mass production of SSLV. Right. So these two things, all of them are important, but these two are having some unique features. So that is why we are paying more attention to it. अब एक ब्रेक लेते हैं, है ना? तो हाँ जी, नहीं आज क्यों खत्म होगा, कल करेंगे. हाँ, थोड़ा ज्यादा स्पीड है, स्लो डाउन करें. ओके, क्योंकि अभी है एरिया ना, मतलब आईटी एंड कम्युनिकेशन एंड देन डिफेंस न्यूक्लियर तो रहता है. और कोई विल स्लो इट डाउन, डोंट वरी. तो कितने देर के लिए? ब्रेक कितने देर के लिए? हाँ हाँ, सिंगल ब्रेक और क्या, खत्म करेंगे. हाँ ठीक है, 20 मिनट चलो. उसकी एक ब्रेक अभी, एक क्लास के बाद. ओके, सो मतलब मिटिगेशन चल रहा है अभी, हम लोग कोशिश कर रहे हैं कि उनको कम करेंगे. इंडिया ने अपने लेवल पे काम किया है कि 2030 तक हम जीरो स्पेस पोल्यूशन मोड में चले जाएंगे. तो रिसेंटली वी लॉन्च्ड वन PSLV, इसमें हमने जीरो स्पेस पोल्यूशन क्रिएट किया. 2030 में हम ये रूटीन बना देंगे. तो लाइक एवरी कंट्री इज डूइंग अ बिट इन पीसेस. तो हर कंट्री अपना अपना कुछ ना कुछ कर रहे हैं. इंटरनेशनली कोई ऐसा रूल रेगुलेशन नहीं है जहां पे कॉन्सेंसस बनाओ कि नहीं हम ये नहीं करेंगे. यस, इंटरनेशनल कमिटीज आर देयर हु आर लुकिंग फॉर दैट हाउ दे कैन मिनिमाइज द स्पेस डेब्री, बट कोई कंपल्शन नहीं है, बाइंडिंग नहीं है कुछ भी. इट्स ऑल अपॉन दैट यू आर एबल टू डू इट, यू विल डू इट. बाइंडिंग कर भी कैसे सकते हैं? आई मीन सोचो, क्लाइमेट चेंज में बाइंडिंग नहीं है, वहां पे भी कॉमन बट डिफरेंशिएटेड रिस्पॉन्सिबिलिटी की बात करते हैं, है ना, कि तुमने ज्यादा कार्बन इमिशन किया है तो तुम ज्यादा पैसे खर्च करो, हमने कम किए हैं तो हम कम करेंगे. पता है ना, बहुत बड़ी पॉलिटिक्स चलती है वहां पे, CBDR की, कॉमन बट डिफरेंशिएटेड रिस्पॉन्सिबिलिटी. तो यहां भी वही कहानी है. आई मीन द डेवलप्ड कंट्री दे हैव पोल्यूटेड अ लॉट इन स्पेस, और अब अगर वो कुछ लेके आ जाएं कि लेट्स नॉट पोल्यूट इट, सो यू आर नॉट बेसिकली गिविंग चांस टू द डेवलपिंग कंट्री टू ग्रो प्रॉपरली, क्योंकि फिर तो आप उसी की आड़ में टेक्नोलॉजी बेचना शुरू कर दोगे. होता तो यही है ना, वर्ल्ड पॉलिटिक्स में एक बहुत बड़ा टूल है दैट फर्स्ट डेवलप्ड कंट्री विल डू समथिंग, दे विल पोल्यूट, दे विल डू कार्बन इमिशन, दे विल डू ओजोन डिप्लीशन, फिर वो कहेंगे कि इससे बहुत खतरा हो गया, फिर वो उसके ऊपर कुछ लेकर आएंगे नई टेक्नोलॉजी, और फिर कहेंगे कि अब ओजोन डिप्लीशन नहीं करना है, ये गैस यूज नहीं करना है. तो कौन सा गैस यूज करना है? तो हमने जो डेवलप किया वो गैस यूज करना है. तो हम बोलेंगे दे दो, बोलेगा नहीं पेटेंट है ना, वो पैसे देने पड़ेंगे तुम्हें. तो फिर इंडिया कहता है कि फिर क्यों पेटेंट है, फिर हम भी करेंगे जैसे तुमने किया 20-30 साल तक, है ना. तो इन सेम वे वी कैन नॉट डू इट इंटरनेशनली. लेकिन हाँ, इंडिया इज नाउ टेकिंग द रिस्पॉन्सिबिलिटी ऑफ बिकमिंग वन ऑफ द स्पेस सुपर पावर. तो हम काम कर रहे हैं, ठीक है. अभी आएगा स्पेस पोल्यूशन टॉपिक है, विल डिस्कस इट. और अभी मैं थोड़ा स्लो चलता हूं, मुझे भी लग रहा था कि आई वाज टू फास्ट. तो हाँ, लग मुझे भी रहा था. नहीं मतलब यू व्हेन यू सी द सेम फेसेस ना, तो आपको भी लगता है कि सब तो पढ़ा ही चुके, आप क्या, राइट. देन आई रियलाइज कि कुछ लोग बाहर भी देख रहे होंगे, बट आई थिंक मोस्ट ऑफ देम आर ऑलरेडी डन द क्लासेस इन ऑफलाइन और ऑनलाइन मोड, बट लेट्स गो अ लिटिल स्लो. तो we talked about this launch vehicles, and in launch vehicles we talked about NGLV, SSLV. These two are important one. RLV is also important. Now when we talk about satellite, right? So satellite की जब बात करते हैं, so there are types of satellite we are using. Let's say when we use remote sensing satellite, then our focus should be on payloads, है ना? तो एक तो payload should be very clear, and we know that if remote sensing satellites are there, the payload is camera or synthetic aperture radar. Okay. So in communication satellite we have multiple transponder. Navigation satellite we have atomic clock. And then we have navigation transponder. And the last one, if we have weather satellite, then we have sounder and imager. Imager is nothing but the camera. We'll talk about that too. So one thing that this should be clear. And now we'll take one by one. So if we go with the remote sensing, so we know that remote sensing means that we explore the chemical and physical nature of an object without physically coming in contact with that, right? So we just throw some waves, or let's say we catch the reflected wave coming out of that object. So reflected wave will come reflected from that object, या फिर ऐसा भी हो सकता है कि हम उसके ऊपर थ्रो करें, है ना? And then we'll measure the change in frequency of that wave. A part of wave will be absorbed and the part of wave will be reflected. Raman effect बताया था मैंने आपको. So this remote sensing is based upon Raman effect or we can also call it a spectroscopy. So it's like throwing some wave and then catch the reflected wave and find out the change in frequency. Every element will make some different change in frequency. So by reading the change in frequency, we can find out which kind of object was there. And this remote sensing that can be of two types. एक है passive one, so passive remote sensing, and the another one is active remote sensing. So when I say passive, it is based on sunlight. Based on sunlight. And payload there will use camera. Because it is based on sunlight, so it can work only in day condition, works only in day and clear weather condition. So if day condition is there, sunlit area is there, then only it will work properly and weather should be clear. Active one is advanced technology and it won't based on sunlight, it is based on microwave. So based on microwave emitted by satellite itself. So microwave will be emitted by satellite. Sunlight तो sun से आएगा, है ना? And payload that which emit this microwave and it will catch the microwave is synthetic. और वो लड़का फिर बोल रहा है 2 महीने बाद कि सर it was a nice experience, my English has improved. वैसे ही होता है. इंग्लिश इज बेसिकली व्हाट, इट्स अ फियर फ्रॉम इंग्लिश. तो नदर्न बेल्ट जो है ना इंडिया का, जो आई मीन स्पीकिंग एंड लिसनिंग, ये गड़बड़ होता है, राइट. और वो इंप्रूव करता है धीरे धीरे. तो बेसिकली प्रॉब्लम इज दैट ओनली, इट्स नॉट लाइक इंग्लिश इज अ प्रॉब्लम. एंड बाय द वे लिसनिंग तो सबकी गड़बड़ होती है. जैसे कि मेरे बच्चे इंग्लिश गाने सुनते हैं और मुझे एक, मतलब जो मेरे पुराने टाइम के जो पुराने गाने, ईगल्स वगैरह, जो उनके गाने सुन लेता हूं मैं, बट जो नए आ रहे हैं जस्टिन बीबर एंड ऑल, आई आई कैन कैच थर्ड लाइन, मतलब दो लाइन समझते समझते तीसरा वो बोल देते हैं. और मेरे बच्चे गुनगुना रहे होते हैं, दे आर लाइक, मैं उनको बिठा के पूछता हूँ क्या बोल रहा है बताओ. एंड वंस यू गेट द लिरिक्स देन यू कैन अंडरस्टैंड. वंस यू रीड इट, या कोई अगर सामने से कह दिया तो समझ में आता है. तो इट्स ऑल डिपेंड अपॉन हाउ एक्सपोज यू आर. क्या चल गया? ओके. तो पैसिव एंड एक्टिव and high resolution multi-purpose data, so it's more useful for defense, so that you can understand in a way that more suitable for strategic use, strategic applications. Although this can also be used in strategic application, but this is better. And if we go with the examples, then in passive, the best example we can have is Cartosat. We are still having lots of Cartosat, 7 to 8 Cartosats are there. And if we go with the example of active one, then we have RESAT, है ना? So RESAT, radar imaging satellite, that's a RESAT. We are having it, right? तो इंडियन अगर रिमोट सेंसिंग सैटेलाइट को हम देखें ब्रीफली, so we have Cartosat, we have ResourceSat, we have OceanSat, and the new version of OceanSat is known as SCATSat. We have ISIS, we have RESAT, and now we have started EOS, Earth Observation Satellites. Out of this 6 category, these 4 are passive. This is active, only one, and this one is new and uniform name of all remote sensing satellite. When I say all remote sensing satellite, means active plus passive. So now ISRO has accepted a new naming system and they call it EOS 01, 02, 03. So now it's by name, you cannot find out whether it is active and passive. By the way, nobody is going to ask you कि अभी EOS 05 छोड़ा है तो एक्टिव है या पैसिव है. ऐसे फैक्चुअल, बिल्कुल फैक्चुअल सवाल नहीं आते, है ना, कि विच सैटेलाइट is what. As a whole, they can frame a question around active and passive, but pinpointed thing, this is the satellite we launched, and then which satellite is that, it won't be. I mean, it's too regressive to go to that level, but we should understand these are the type of satellite we have. Cartosat, in a one word, it is for map making. Mapping, you can say, not map making, mapping. So mapping for all purpose, rail development, railways development, roadways development, urban development, rural development. So wherever the mapping is there, we'll use Cartosat. Cartography is mapping only. ResourceSat is management of natural resources. Natural resources. Now, how these things will be different? It's all that which kind of camera we are having. So even the cameras are of different types. So we have different type of camera, like let's say in cartoon, we use a camera that is called panchromatic camera. Panchromatic camera. तो ये क्या करता है, पैन कहते हैं, पैन भी कहते हैं, PN. तो ये हाई कॉन्ट्रास्ट कैमरा है, ये लाइट कलर और डार्क कलर को कॉन्ट्रास्ट में दिखाएगा. इट वोंट गिव यू डिटेल्स, रादर इट विल गिव यू हाई कॉन्ट्रास्ट. तो हाई कॉन्ट्रास्ट से मैपिंग अच्छी होगी, राइट? So that is why it is used in mapping. So mapping is not like, you just map the things according to the landforms and everything. Resource sat, ocean sat, they have different type of camera, LIS, LISS or WIFS, जो उनके अलग अलग peculiarity है. So accordingly कैमरा के हिसाब से उनका काम होगा. This is for oceanic and coastal area. So if you want to do anything in oceanic and coastal area, management, early warning, surveillance, then we'll do it. This is important. This is called hyperspectral imaging satellite. And when it is hyperspectral imaging satellite, then it can pinpointedly find out some information, right? So it can focus on a particular object or area. So out of lots many things, if you want to focus on a particular object or area, you can use this HISES, like for mineral exploration, है ना? This is का best application है mineral exploration. Defense application में भी काम आता है. The particular mineral you need to find it out, we can use it. And reset, we know the story of the reset, we have talked about in the classes and all, that India launched it so that India can have better remote sensing capability in the border areas to stop the infiltration of the terrorist. But we never said that. We never mention it. Although we know it that we are using it for this purpose too, but then reset is beyond only this, spying and other thing. तो मेनली हम इसको करते हैं खरीफ क्रॉप इवैल्यूएशन के लिए और डिजास्टर मैनेजमेंट and flood evaluation and all. So this is active and kharif crop evaluation, disaster management, flood evaluation, and a strategic thing. So by name, chances are very less that they can ask, but yes, these three are the important one and the current one. ये सब तो धीरे धीरे phase out हो रहा है. So if they have to ask something about remote sensing, they might take the name of EOS. So one thing we should know that when they ask about EOS, they are basically entering into the remote sensing. ISIS or reset, active or strategic मामले में हो जाएंगे, है ना? So one thing that you should be knowing, the basic science of remote sensing and application of it. Like they have asked one question in remote sensing in the past. उसमें यही था कि remote sensing can be used for 4 ऑप्शन थे. उसमें था कि mineral exploration, mapping, urban development और traffic management. उसमें था कि कौन कौन से हैं. तो traffic management छोड़ के बाकी सब थे. Because managing traffic is not a function of remote sensing. That's a function of navigation satellites. Because remote sensing satellites never be at one particular place and giving you the live feed. वो CCTV camera नहीं है. Remote sensing satellite is always constantly moving. So when it is constantly moving, so it won't give you the real time image. What you get is an image that was shot and clicked, recorded at least few hours back. तो कम से कम 6 घंटा पहले का वो इमेज है. तो क्या ट्रैफिक मैनेजमेंट करेंगे आप? ऑल्दो इंडिया ने एक अटेम्प्ट किया था और अटेम्प्ट ये था अटेम्प्ट ये था कि इंडिया ने कहा था कि हम एक रिमोट सेंसिंग सैटेलाइट लगाएंगे कहाँ पे, जीएसओ में. So GSO, geostationary orbit, and there will put remote sensing. और हमने लॉन्च भी किया था बाय द वे, बट वो मिशन फेल कर गया. राइट. So it was one of its own kind, where a remote sensing satellite will be placed in GSO, 36,000 kilometer away, and then this will focus on a particular part of the India or area. It will record the images from there, and it can give you the live feed. And the purpose was that it will help in better disaster management. But 4 साल पहले हमने लॉन्च जब किया तो that mission failed because of the failure of launch vehicle. So we could not do it. It was a very costly satellite because obviously the camera and equipments were very powerful, so that it can record the images from 36,000 kilometer to. अब दोबारा हमने कुछ भेजा नहीं है, वो मिशन फेल कर गया था. राइट. सो फेल कर गया इसलिए उस समय तो पढ़ते थे बट अब नहीं पढ़ते. बट यस, you can have the live feed, you can put a CCTV camera, but for that you have to put the satellite into geostationary orbit, and for that you have to develop it. अगर इंडिया ने एक डेवलप किया, फेल कर गया, तो ये बात तय है कि पाइपलाइन में कोई ना कोई होगा. अभी तक तो लॉन्च नहीं किया है. In a coming time we might see that we are having a satellite, remote sensing satellite in GSO, right? But till now we don't have anything in that way. Anyway, similarly we'll explore satellite communication. Satellite communication. And satellite communication में simple है that we use telemetric capacity of the satellite so that we can, transfer the signals to different locations like DTH and all. तो एक satellite जो communication में वो क्या कर सकता है, तो it can take the signal, and that is called uplink. It can amplify it. यहाँ पे क्या होगा, amplification of the signal, and then this signal will be given back. So signal is going, it is being amplified, it is coming down, then it is called downlink, and this downlink will be important, right? Because this downlink can be done at the different places, anywhere in the world, right? So we have two types of broadcasting you need to understand. One broadcasting is your radio broadcasting. For radio broadcasting we don't need any satellite system, right? Because radio broadcasting के लिए ionosphere है, right? So ionosphere, and radio wave will go strike onto ionosphere, it will come back. So radio wave will go and come back, right? Why? Because radio wave has got very low frequency. Frequency means energy, है ना? एक टॉपिक में अभी आपको बता देता हूं, मतलब जनरल साइंस का टॉपिक है बट बहुत इंपॉर्टेंट है ना? वो नॉर्मली क्लासेस में नहीं होती, लेकिन ठीक है, वी हैव एक्स्ट्रा टाइम, देन वी कैन टॉक अबाउट इट, एंड दैट विल गिव यू द आइडिया ऑफ द एंटायर वेव दैट वी आर यूजिंग, राइट? ठीक है, तो अभी हम लोग थोड़ा डी टूर करते हैं और देखते हैं. एक तो बहुत इंपॉर्टेंट टॉपिक है, दैट इज ईएम वेव. बिकॉज अभी के पॉइंट पे तो मैं ये भी नहीं कह सकता ना आपको एनसीईआरटी से पढ़ लेना, है ना? बट व्हाट आई फील, एंड not only feel, rather there is a track record that they have asked question from these area. EM wave, very important, because EM wave is nothing, it's applied science, you know, it's a science, core science, but EM wave we apply at various places, right? So electromagnetic wave or EM wave, right? We call it electromagnetic wave. So we have two types of wave in wave physics. One is called mechanical wave, sound is mechanical wave, and another is electromagnetic wave. If a wave is mechanical wave, it will always require a medium to travel, right? Because mechanical wave means the particle will oscillate. Particle होना चाहिए, particle ही move करेगा, तो particle ही move करेगा तो medium में ही तो move करेगा. पार्टिकल होने के लिए मीडियम होना जरूरी है. दैट इज वाई साउंड वेव इज मैकेनिकल वेव. इफ देर इज नो एयर इन दिस रूम द साउंड विल नॉट ट्रेवल. फिर ट्रेवल करेगा दीवार से होकर, कि दीवार मीडियम है. दैट इज वाई वी आर नॉट एबल टू हियर एनीथिंग इन द स्पेस. इफ टू पीपल आर देयर ऑन मून, स्टिल दे विल टॉक टू ईच अदर बाय रेडियो सेट्स. तभी भी बात कैसे करेंगे? रेडियो सेट से ही करेंगे. वो डायरेक्टली बात नहीं कर सकते, है ना. EM वेव डजंट रिक्वायर मीडियम. ये मीडियम इसके लिए नहीं चाहिए, है ना. तो EM wave is nothing but a pure energy, and then we have different types of EM wave, and we can make an arrangement. जैसे एक है रेडियो वेव, फिर इसके आगे बढ़ते हैं तो माइक्रोवेव, फिर इसके आगे बढ़ते हैं तो इंफ्रा रेड, देन वी कम टू विब्जियोर, उल्टा लिखता है. देन व्हेन वी गो टू देन अल्ट्रावायलेट, then X-ray, and then the last one is gamma rays. Now this is something जो इसी तरीके से लिखना है पहले तो, because if you read in that way, then you will realize one thing, that we have arranged everything. आप देखो एक एरो भी तो है ना, there is a direction we have maintained, रेडियो, माइक्रो, इंफ्रा. याद करना बहुत आसान है. पहले विब्जियोर उल्टा लिखो, रेड क्या पहले क्या लिखो? इंफ्रारेड. वायलेट के बाद क्या लिखो? अल्ट्रावायलेट, है ना. फिर एक्सरे और गामा रेस, राइट. इधर फिर पहले रेडियो फिर माइक्रो, राइट. एंड इफ यू मेंटेन दिस देन यू रियलाइज वन थिंग. आप क्या रियलाइज करते हो? कि इसका जो वेवलेंथ है दैट इज डिक्रीजिंग. वेवलेंथ इज एवर डिक्रीजिंग. इस पूरे सीरीज में वेवलेंथ क्या हो रहा है? कम हो रहा है. हम पढ़ते हैं ना बचपन में कि लाल का वेवलेंथ सबसे ज्यादा होता है, तभी ये ज्यादा दूर तक जाता है, इसकी स्कैटरिंग कम होती है. तो देखो, इन ऑल 7 colors red has got the maximum wavelength because wavelength is decreasing. Violet has got the minimum wavelength. Relationship of wavelength with frequency is reverse. अगर वेवलेंथ ज्यादा है तो फ्रीक्वेंसी क्या होगी? बढ़ती चली जाएगी. So it will have more frequency, right? So आप बताओ, सबसे ज्यादा फ्रीक्वेंसी किसकी? गामा रेडिएशन की. सबसे कम किसकी? रेडियो वेव की. Frequency means what? Frequency means how many times the wave is striking on a particular thing. How many times it's oscillating. If I just tap once in 1 second, तो फ्रीक्वेंसी 1 हर्ज की है कि नहीं? अगर मैंने 1 सेकेंड में इस मार्कर से इस बोर्ड पे एक बार मारा, तो आप कह सकते हो कि मेरी फ्रीक्वेंसी अभी जो है 1 हर्ज है. अगर मैंने 1 सेकेंड में 2 बार 2 हर्ज, 1 सेकेंड में 500 टाइम्स तो 500 हर्ज. Just how many time it vibrated, how many time it struck upon. अब सोचो कि अगर एक बार में एक बार मारा तो कोई एनर्जी तो गई होगी ना, x अमाउंट ऑफ एनर्जी. लेट्स कंसीडर 1 सेकेंड में 2 बार मैंने मारा तो 2x एनर्जी. 1 सेकेंड में 50 टाइम्स स्ट्राइक किया तो 50x एनर्जी. यू कैन गेट माई पॉइंट. तो फ्रीक्वेंसी और एनर्जी एक मान लो, राइट. इट्स लाइक दैट ओनली. एंड क्लियर कट ऐसे याद रखना, जैसे बोला कि number of strikes in 1 second on this board by this marker is frequency. Similarly, number of vibration occurring in 1 second in a wave is frequency. If number of a strike is increasing, then energy will increase. है कि नहीं? और बहुत अगर मान लो कि 1 सेकेंड में मैंने यहां पे अगर 5000 बार मार दिया तो छेद भी हो जाएगा, होगा कि नहीं? That is called penetration power. So more energy, more penetration. You get my point? So here what will happen? अब देखो, तो एनर्जी भी क्या हो गई इसकी? बढ़ गई. और पेनिट्रेशन भी क्या हो गया? पेनिट्रेशन बढ़ गया. अब सब क्लियर हो जाएगा. अब आप बताओ, गामा रेडिएशन इज मोस्ट डेंजरस वन. व्हाई? लॉजिक यहीं से निकले. बिकॉज फ्रीक्वेंसी हाई, एनर्जी हाई, पेनिट्रेशन हाई. इट इज सो पेनिट्रेटिव दैट इट कैन कवर करके और कहां पहुंचेगा? बोन मैरो में. इन बोन मैरो इट कैन मेक सम चेंजेस इनटू द सेल, और वही सेल के डीएनए में, बिकॉज इट इज हैविंग हाई एनर्जी एंड हाई पेनिट्रेशन वैल्यू. दैट इज व्हाई गामा रेडिएशन से बचना बड़ा मुश्किल होता है. इफ यू वांट टू हाइड योरसेल्फ फ्रॉम गामा रेडिएशन, you need a barrier of concrete and steel. which should be minimum 20-25 feet thick. इसे लोग कहते हैं ना कि अगर न्यूक्लियर एक्सप्लोजन हो जाए तो बचने का क्या चांस है? बेसमेंट. बेसमेंट इज ओनली प्लेस जहां पे चांसेस है कि बच सकते हैं. दिस वॉल एवरीथिंग वोंट बी एबल टू स्टॉप. इसमें तो आराम से गामा रेडिएशन आएगा, जैसे चाय छानते हैं ना वैसे आएगा छन छन के, और बढ़िया प्योर, है ना. अब बेसमेंट की जो वॉल होती है वो 25 फीट मोटी तो नहीं होती, लेकिन बेसमेंट के चारों तरफ क्या होता है ना, लैंड ही तो होता है. that the land and wall that will create a kind of barrier, but still ऊपर से आएगा. So it's kind of a little bit of protection can be there, right? तो अब ये now you have to understand this. Wavelength is low, उससे कोई फर्क नहीं पड़ता. As we go from there, energy, frequency and penetration power is increasing. तो सबसे कम penetration किसकी है? रेडियो की. That is why radio wave is not able to. ओके तो आप देखो रेडियो वेव आयनोस्फेयर से टकरा के वापस आएगा, बिकॉज इट वोंट हैव एनर्जी एनफ टू क्रॉस द आयनोस्फेयर, है ना. एंड दैट इज रेडियो वेव, बिकॉज ऑफ इट्स लो पावर, लो एनर्जी. हाँ जी, वो पूरा ही आयनोस्फेयर मान लो, तो इट विल बी रिफ्लेक्टेड बाय ionosphere, right? So it will be used in radio broadcasting, and that is why radio broadcasting never required satellite system. Ionosphere is working as a reflector of the radio wave, right? Microwave क्या है ना, माइक्रोवेव की पेनिट्रेशन पावर इज बेटर देन रेडियो वेव. एनर्जी लेवल is high, so it will cross the ionosphere. And when it is crossing the ionosphere, then what will happen? You will have to put a reflector, and satellite is a reflector only. So the purpose of satellite is nothing but to reflector, right? But reflector है तो थोड़ा सा काम भी ले लेते हैं, और हम कहते हैं कि ठीक है तुम्हें हम वीक देंगे, वीक सिग्नल देंगे, एम्पलीफाई करके भेजना, मतलब उसकी पावर बढ़ा देना, ठीक है, because it's a satellite. It will take up its uplink, it can amplify it, and it can send it back, right? It can send it anywhere. For that matter, ionosphere से भी कहीं भी भेज सकते हैं आप, है ना. तो माइक्रोवेव टीवी ब्रॉडकास्टिंग and microwave can penetrate ionosphere, and that is why satellite communication satellites are used as reflector. So communication satellite are used as reflector, right? That's microwave. And microwave बहुत इंपॉर्टेंट इसलिए भी है कि देर आर डिफरेंट एप्लीकेशंस ऑफ माइक्रोवेव अपार्ट फ्रॉम टेलीविजन. आई मीन कम्युनिकेशन सिस्टम्स माइक्रोवेव पे ही चलते हैं. जैसे वाईफाई रेडियो वेव माइक्रोवेव पे चलता है, रेडार सिस्टम माइक्रोवेव पे चलते हैं. योर मोबाइल फोन्स आर रनिंग ऑन माइक्रोवेव, बिकॉज वेन एवर वी टॉक अबाउट मोबाइल फोन इट्स हेल्थ कंसर्न, तो आप देखते हो ना माइक्रोवेव ही लिखा हुआ आता है. तो यहाँ पे वाईफाई चल रहा है आपके माइक्रोवेव पे, आपका ब्लूटूथ चल रहा है माइक्रोवेव पे. हाँ जी, वाईफाई तो लाइट से चल रहा है ना, राइट. फिर रेडार सिस्टम इज माइक्रोवेव, राइट. मोबाइल टेलीफोनी is microwave, and one of the very important feature is microwave oven, right? So microwave oven, you know that, but कई बार लोग कहते हैं ना microwave is harmful for the human body and all. It doesn't hold any truth in a proper way. WHO ने कुछ नहीं कहा अभी तक. WHO ने ये कहा कि अभी तक जितना भी पढ़ा है उसके हिसाब से कोई भी हार्म तो नहीं नजर आता है. WHO ने इस पे भी नॉट 100% affirmative, not 100% negative. बीच बीच में कि studies that has been done till date is not suggesting that microwave can cause brain tumor or brain cancer. इसका मतलब है कि अभी तक स्टडी में पता नहीं चला, आगे का पता नहीं है कि नहीं, पर ठीक है, नहीं होगा। कारण है कि वी आर यूजिंग वेरी लो पावर माइक्रोवेव। एक तो एनर्जी लेवल बहुत कम है, एंड इट्स नॉट लाइक दैट मोबाइल फोन यहां है तो माइक्रोवेव तो है ही ना हमारे आसपास। When this area is covered with the Wi-Fi, it is covered by the microwave. When you are there at near airport, you are enveloped by the microwave. तो है ही। तो एक होता है एनर्जी, दूसरा होता है पावर। पावर इज एनर्जी अपॉन टाइम, राइट? तो इवन इफ समथिंग इज हैविंग लो एनर्जी, यू कैन हैव द मोर पावर, है ना? तो लो एनर्जी से भी पावर हम ले सकते हैं। तो माइक्रोवेव ओवन इज बेस्ड ऑन व्हाट? इट इज बेस्ड ऑन हाई पावर माइक्रोवेव। Energy of the microwave is low, but per unit time, how much energy you are giving, that will create what? Power. है कि नहीं? तो माइक्रोवेव oven is around 1000 watt माइक्रोवेव, और 1000 watt माइक्रोवेव का मतलब होता है 960 के आसपास होता है। मतलब कि दिस इज हाई पावर माइक्रोवेव, एंड वंस यू पुट एनी फूड आर्टिकल इनटू द माइक्रोवेव, तो उस फूड आर्टिकल में जो हाइड्रोजन के एटम होंगे, वाटर की मॉलिक्यूल्स होंगी, वो एनर्जी को एब्जॉर्ब करेगा, वो वाइब्रेट करेगा, and the vibration will be too vigorous that it will start emitting heat. So the water molecule inside the food article, that will become hot because of lots of energy being absorbed by it. तो खाना अपने आप गर्म हो जाएगा, है कि नहीं? So that is called microwave cooking, right? एयर फ्रायर तो हॉट एयर को, एयर फ्रायर इज नथिंग बट इट्स अ ओवन, छोटा ओवन है जिसमें नाइक्रोम के वायर लगे हैं। नाइक्रोम का वायर जो हीटर का वायर होता है, जो इमर्शन रॉड में होता है या हीटर में होता है, तो नाइक्रोम वायर विल बी रेड, और फिर फैन लगा हुआ है, उस फैन को छोटे से जगह में घुमाता है हवा को, गर्म हवा को। तो एयर फ्रायर में खाना आपको लगता है कि बहुत जल्दी हो गया एज कंपेयर टू ओवन, बिकॉज ऑफ जस्ट वन थिंग, इट्स अ वेरी स्मॉल कंपार्टमेंट, राइट? ओवन इज बिग वन। ओवन को रेडी होने में भी लगेगा 5 मिनट, उसको प्रीहीट करना पड़ता है। माइक्रोवेव ओवन में प्रीहीट 30 सेकंड में हो जाता है, राइट? सो इट्स अ मिनिएचर ओवन, नथिंग एल्स। एनीवे, अभी आपने देखा होगा ना कि इंडिया ने एक लेजर गन निकाला है ना, टू शॉट डाउन द एरियल टारगेट्स लाइक ड्रोन्स एंड ऑल। अभी इंडिया ज्वाइंड द लीग ऑफ द नेशन व्हिच हैज गॉट सच साइंस फिक्शन मूवी वाला गन। तो ऐसा नहीं है कि इंडिया ने पहली बार ऐसा किया हुआ है। इंडिया के पास माइक्रोवेव बीम वेपन भी है, एक्सरे बीम वेपन भी है, और लेजर वाला भी। लेजर वाला तो था ही, वही उसको दुर्गा कहा था पहले। तो देयर इज समथिंग कॉल्ड काली, है ना, किलो एम्पियर लीनियर इंजेक्टर डिवाइस, दैट्स वर्क्स ऑन माइक्रोवेव। 5000 किलोवाट का माइक्रोवेव बीम छोड़ते हैं। अब सोचिए 100 वॉट माइक्रोवेव से खाना पक जाता है कि नहीं? मतलब खाना इतना गर्म हो जाता है कि आपकी उंगली जल जाए। अगर इस मोबाइल फोन को माइक्रोवेव में डाल दो और चला दो, तो फिर कभी दोबारा स्टार्ट नहीं होगा। एंड दैट इज ओनली वन। इंडिया ने जो बीम वेपन बनाया वो कितना का है? 5000 किलोवाट। तो 5000 मल्टीप्लाइड बाई 1000 वॉट। तो सोचो उतना अगर हाई पावर माइक्रोवेव बीम अगर हम छोड़ रहे हैं, एक तो बहुत दूर तक जाएगा। सेकेंड, इफ अ ड्रोन और एनी एरियल टारगेट विल कम इनटू द कांटेक्ट ऑफ दिस माइक्रोवेव, तो उसके इलेक्ट्रॉनिक्स बिल्कुल खत्म हो जाएगी और वो गिर जाएगा। तो वी हैव डेवलप्ड अ डिवाइस हियर दैट इज कॉल्ड काली 5000, एंड काली 5000 इज बेस्ड ऑन माइक्रोवेव। माइक्रोवेव पे, और एक जो लेजर वाला बनाया है उसका नाम दुर्गा है, जिसको अभी हमने टेस्ट भी किया है। अब तो दुर्गा कोड नेम होगा। तो काली 5000 is microwave weapon. Then we come to infrared. Infrared ठीक है, infrared is good for in a space tech. If we talk about, we do use infrared. जो HISIS है ना आपका, HISIS remote sensing satellite, उसमें हम infrared यूज करते हैं। infrared का मतलब होता है anything which is hot will emit infrared, right? Night vision camera is based on infrared. थर्मल कैमरा जिसको कहते हैं वो भी होता है किस पे बेस्ड? इंफ्रारेड। So is there anything in this area which are having some temperature, then it will emit infrared. जितना ज्यादा टेम्परेचर होगा इंफ्रारेड इमिट करेगा. सिंपल सी बात समझो आप, एक सिंपल फिजिक्स है. अगर किसी चीज को गर्म करते हो, लेट्स से इफ देयर इज अ मेटल पीस एंड इफ आई पुट दिस मेटल पीस ऑन फ्लेम, तो गर्म तो होगा. और अभी हमने छोड़ दिया इसको 10 मिनट के लिए फ्लेम के ऊपर ही, और जब बहुत गर्म होगा तो ये ब्लैक कलर का मेटल पीस कैसा दिखेगा? रेड दिखेगा. तो रेड दिखना शुरू हो गया. रेड दिख रहा है. व्हेन इट इज रेड हॉट, तो आप कहते हो बहुत हॉट है. आप कह रहे हो बहुत हॉट है बिकॉज यू कैन सी द रेड लाइट कमिंग आउट ऑफ इट. यू गेट माय पॉइंट, राइट? तो यू आर सेइंग इट इज वेरी हॉट बिकॉज यू सी दैट रेड लाइट इज कमिंग आउट ऑफ इट. अब इसको उल्टा समझो, ये बहुत हॉट होगा तो रेड लाइट निकालेगा, है कि नहीं? अगर हम इसको और गर्म करते जाए तो पता है क्या होगा? वो इधर शिफ्ट करेगा. एनर्जी लेवल बढ़ रही है ना, उधर ही जाएगा. अगर ये अब बहुत गर्म करेंगे तो इट वोंट बी रेड, रेदर इट वोंट बी इन दिस स्ट्रक्चर. ये पिघल जाएगा. पिघलने के बाद लोहा रेड नहीं दिखता, येलो दिखता है. अगर आपने कभी देखा हो टीवी वगैरह में मोल्टेन आयरन, इट्स नॉट रेड. रेड विल बी देयर until the structural integrity is there, maintained. तो रेड है. द मोमेंट इट मेल्ट्स इट विल बिकम येलो. येलो के आगे ग्रीन तक नहीं जाएगा, डोंट वरी, बिकॉज उससे पहले वेपराइज हो जाएगा. बट अगर आप गैस यूज करते हो तो गैस में देखो जो लैंप निकलती है ना गैस की, वो किस कलर का होता है? ब्लू. अगर वो येलो हो जाए तो अच्छा टेम्परेचर है या बुरा टेम्परेचर है? येलो अगर निकल रहा है इसका मतलब फ्यूल बर्न ठीक से नहीं कर रहे, and heat is less. Blue has got the maximum heat, maximum energy. So as we go towards this red, yellow, blue, and a blue flame is the highest temperature flame. If you get the blue flame out of the burner, it means that your fuel is burning very well. It is giving you lots of temperature, maximum temperature that it can give. है ना? इसके बियॉन्ड तो आप जा भी नहीं पाएंगे. तो सिमिलरली बहुत गर्म है तो रेड हॉट है, तो बहुत गर्म से थोड़ा कम है तो क्या है? उसे क्या निकल रहा होगा? इंफ्रारेड. यू गेट माय पॉइंट? तो लेट्स से अगर ये 200 डिग्री पे रेड हॉट होता है तो 150 डिग्री पे क्या हो रहा होगा? इंफ्रारेड निकल रहा होगा. आप ये भी कह सकते हो इंफ्रारेड है ये, या इंफ्रारेड हॉट है, बट इंफ्रारेड हम नहीं देख सकते. वी कैन सी रेड, वी कैन ओनली सी इन दिस रेंज. तो टू इंफ्रारेड यू हैव टू पुट सम इंस्ट्रूमेंट. इंस्ट्रूमेंट कुछ भी हो सकता है, स्पेक्ट्रोस्कोप, स्पेक्ट्रोमीटर, एंड देन दैट इज व्हाट वी कॉल इट थर्मल विजन, है ना? थर्मल विजन में क्या है कि दो चीजें हैं, ये 20 डिग्री पे है, ये 30 डिग्री पे. तो 30 डिग्री वाला इंफ्रारेड निकालेगा, 20 डिग्री वाला भी इंफ्रारेड निकालेगा, लेकिन इसकी फ्रीक्वेंसी इंफ्रारेड की क्या होगी? ज्यादा होगी, इसकी कम होगी. तो दूर से हम क्या करेंगे? इंफ्रारेड की फ्रीक्वेंसी देखेंगे और फ्रीक्वेंसी को कन्वर्ट करेंगे to get to know about the temperature of these two objects accurately. That is also remote sensing. समझ में आ रही है? अब इसी को आप कहो तो रिमोट सेंसिंग कह दो, इसी को चाहो तो कह दो थर्मल विजन. और सेम टेक्नोलॉजी वी यूज्ड ड्यूरिंग कोविड. कोविड में हमने एक बहुत बड़ा पैराडाइम शिफ्ट देखा, और वो क्या था? द एंटायर वे टू मेजर द बॉडी टेम्परेचर चेंज्ड. पहले माउथ में थर्मोमीटर लगाते थे, थर्मोमीटर ही बदल गया. Now we have infrared thermometer, right? Because anything which is hot will emit infrared, and if something is hotter, the frequency of infrared will keep on increasing. है कि नहीं? तो हम लोग भी तो हॉट है, मतलब टेम्परेचर वाला हॉट. 37 डिग्री पे है ना हम लोग. So our body at this particular time is emitting a particular frequency of infrared that is equal to 37 डिग्री. अब फीवर लग गई तो 38 हो गया, तो फ्रीक्वेंसी बढ़ गई. सो दैट गन, दैट हैंडल डिवाइस टू मेजर द टेम्परेचर इज इंफ्रारेड थर्मोमीटर. वो क्या करता है? बॉडी से इंफ्रारेड निकल रही है, उसको वो कैलकुलेट के टेम्परेचर में दिखा दे रहा है. सो ऑल थर्मल एंड ऑल काइंड ऑफ. अब देखो ये क्या है ना, दिस, अब ये थोड़ा सा मैं डिटूर कर गया, बट इट्स इंपॉर्टेंट वन, राइट? ऑल्दो दैट इज वाई समटाइम पीपल आस्क ना कि जनरल साइंस में पढ़ना क्या. हम जनरल साइंस में क्या पढ़ना शुरू कर देते हैं? हम पढ़ते हैं न्यूटन्स लॉ, फर्स्ट लॉ, सेकंड लॉ, थर्ड लॉ. इट्स ओके, यू हैव टू गो विद दैट, The most important thing, UPSC will rarely ask Newton's law, but what they ask, they can ask things related to Newton's law. है कि नहीं? पढ़ रहे हैं EM webs, but the focus should be where? The application part. Right. अब Doppler effect पढ़ रहे हैं. Radar is working on Doppler effect. Right. So Doppler effect, in theory, it's not important, but what kind of applications are there with the Doppler effect, where all we apply over there, that is important. Anyway, तो हाँ जी, नाइट विजन में क्या है कि मान लो कि मैं खड़ा हूं, माई बॉडी टेम्परेचर इज 37, इट्स टोटली डार्क, तो अर्थ का एवरेज टेम्परेचर तो मेरे बॉडी से तो कम ही होगा, या फिर ज्यादा भी हो सकता है कि नहीं? जो हम कहते हैं ना 42 डिग्री है बाहर, वो तभी है जब सनराइज बहुत स्ट्रांग है, अदरवाइज ठीक है, क्योंकि स्ट्रक्चर गर्म हो जाते हैं ना, अर्थ गर्म हो गया, दीवारें गर्म हो गई, इसको हम कहते हैं ना अर्बन हीट आइलैंड बन जाता है ना. बट इफ यू गो टू द वाइल्ड एंड दोज एरियाज वेयर नाइट विजन कैमरा रिक्वायर्ड, तो चांसेस आर वेरी हाई एंड 99% केसेस व्हाट विल हैपन, योर बॉडी टेम्परेचर विल बी 37, सराउंडिंग टेम्परेचर वुड बी अराउंड 22, 25, 26, 27, 30 डिग्री, है कि कुछ कंडीशन ऐसी होंगी जहां पे ऐसा भी हो सकता है कि सराउंडिंग इज 55 डिग्री और 50 डिग्री, यू आर 37 डिग्री, स्टिल यू आर हैविंग टेम्परेचर डिफरेंस. तो मान लो ये दीवार है 40 डिग्री पे, मैं 37 डिग्री पे, अंधेरा है, तो दीवार से इंफ्रारेड जो आ रही है वो तो आ रही है 40 डिग्री वाली, फ्रीक्वेंसी हाई है, लेकिन मेरे बॉडी से इंफ्रारेड जो आ रहा है वो कितना? 37. तो माई कटआउट विल बी देयर, you will just get an impression that human like a structure is there, because this particular area, my body, that body is emitting infrared of 37 degree. So frequency of infrared coming out of my body and frequency of infrared, क्योंकि या तो मेरे कॉन्टेक्स्ट में सराउंडिंग ठंडा होगा, नहीं तो गर्म होगा, है कि नहीं? इसलिए आप देखोगे ना कि जो इंफ्रारेड से बचने की कोशिश करते हैं, या तो रिफ्लेक्टर लगाते हैं, इट्स लाइक शाइनी फॉयल लाइक, और वो क्या करते हैं, वो कहते हैं कि बहुत सारा मिट्टी डाल लो बॉडी पे. तो बहुत सारा मिट्टी डालने का मतलब क्या होता है? यू जस्ट ट्राई टू, आई मीन, कूल डाउन योर बॉडी. को रोक नहीं सकते आप, क्योंकि मिट्टी सूखेगा फिर से इंफ्रारेड निकलेगा. टिल द टाइम दिस, आई मीन, थिक मिट्टी एंड दिस ऑल that mud kind of thing is there on body, it will cool down your body. So the body is cooled down, it means that you will merge with the environment. उसमें भी chances बहुत कम. That is why this night vision equipments are there with the army, because उनको डील करना पड़ता है, right? Anyway, चलो इससे आप आगे बढ़ते हैं. ये तीनों, इसको भी देख लेते हैं एक छोटा सा. ये तीनों को कहते हैं आयनाइजेशन रेडिएशन. आयनाइजेशन रेडिएशन, ये सीधा डीएनए डैमेज करता है, and if DNA damage is there, then it can turn into cancer. Now you understand why it is called ionization radiation, because of high energy. The energy level is very high. As we go, energy is high. Ultraviolet will have 3 types of ultraviolet: A, B and C. C एक ओजोन रोक लेता है. A, B, C में A की एनर्जी सबसे कम, B की मिडिल, C की सबसे ज्यादा. अल्ट्रावायलेट C की एनर्जी बस समझो कि एक्सरे के आस ही पास. Either you get exposed to the X-ray or you get exposed to ultraviolet C, it will have the same impact. But the best thing is that the stratospheric ozone will block and absorb the ultraviolet C. अगर अल्ट्रावायलेट C आ रहा होता तो अर्थ पे लाइफ ही नहीं होती, क्योंकि कुछ आयनाइज हो जाता, मतलब इतना केमिकल डिग्रेडेशन करता, कुछ बनता ही नहीं. ए पहुंचता है, कोई प्रॉब्लम नहीं. ए से ज्यादा क्या होगा? टैनिंग होगी. बी पहुंच जाते हैं 15-20% तक. एंड अल्ट्रावायलेट बी कैन डैमेज यू, because it may cause skin burn, it may cause cataract, and it may cause, in some cases, if body is not having proper melanin, it may cause cancer. सी कैन श्योरली कॉज कैंसर, लेकिन वो आता नहीं है, ये अच्छी बात है ना। और उस हिसाब से एक्सरे और गामा रेडिएशन देख लो आप, राइट। अगेन एक्सरे में ध्यान रखना, एक्सरे इन जनरल इट्स कार्सिनोजेन। Our X-ray can be used in medical technologies. तो कहते हैं रेडियोग्राफी या फिर सीटी स्कैन। रेडियोग्राफी या सीटी स्कैन, इंडिया में तो एक तो प्रॉब्लम ये भी है ना कि हम अनसाइंटिफिक तरीके से बात करते हैं। हम कहते हैं एक्सरे करवाने जा रहे हैं, मेडिकल वाले उस पर भी लिखा होता है एक्सरे होता है ना। इट्स अ रियल वर्ड इज रेडियोग्राफी। तो रेडियोग्राफी इज द थिंग। इन रेडियोग्राफी वी यूज एक्सरे, इन सीटी स्कैन कंप्यूटर टोमोग्राफी वी यूज एक्सरे। लेकिन वो एक छोटा सा एक्सरे का पल्स होता है माइक्रोसेकंड के लिए, एंड लो पावर एक्सरे इज देयर। तो चांसेस ऑफ गेटिंग डैमेज बाय दिस एक्सरे इज वेरी लेस, नेग्लिजिबल नहीं है, तो नहीं है। जो अच्छे मेडिकल प्रैक्टिशनर होते हैं वो बोलते हैं कि मल्टीपल एक्सरेज नहीं होने चाहिए। मतलब ठीक है, वंस इन अ वाइल, पूरे लाइफ टाइम में 10-15 एक्सरे हो गए बहुत है। और लाइफ टाइम में 10-15 एक्सरे हो रहा है तो आपकी लाइफ बहुत इवेंटफुल है, इसका मतलब है ना। अदरवाइज एमआरआई इज वेरी गुड वन, क्योंकि एमआरआई फिर क्या यूज करता है, वो मैग्नेटिक रेजोनेंस इमेजिंग टेक्नोलॉजी। एनीवे, तो वी जस्ट wanted to explain one thing, that how the satellite communication is important and why satellite communication satellite should be there. So now we understand satellite communication satellites are there at 36,000 kilometers, way above the ionosphere, and this satellite has just one role to play, that is of reflector of microwave, right? And then we send the signal, uplink it, it will amplify, it will link it down, है ना। अब देखिए communication satellites of India. So there, एक तो है, पहले इनसेट के नाम से छोड़ना शुरू किया था। इनसेट वाज ओल्डर एंड इट वाज कम्युनिकेशन प्लस वेदर। सो इनसेट यूज्ड टू बी बोथ कम्युनिकेशन एंड वेदर, दोनों सैटेलाइट, राइट। देन द सेकेंड सीरीज वी स्टार्टेड विद द नेम ऑफ जीसैट। जीसैट is old, not older, but old, and it is only communication. There is nothing else that we have added. And now we have CMS, present. So present generation of satellite that we are having is CMS, and it is again only communication. Only communication. Right. So again, if you go, then there was a time when India started communication satellite. So our communication need was limited. Rather, we wanted to get into the communication and weather both. But India was not having the capacity to build up their own satellite. Launching capacity भी नहीं थी, तो हमने अमेरिका से सैटेलाइट बनवाया, उन्हीं से लॉन्च करवाया। तो हमें चाहिए था मल्टीपर्पस, लेस मनी मोर वर्क, राइट। 1983, तो 1983 का इंडिया आप समझ ही सकते हैं, इट वाज नथिंग, राइट। सो अराउंड दैट टाइम in 1983, this was okay to have communication and weather in one satellite only. But 2002 onwards, we started using dedicated communication satellite, और 2020 में हमने उसको नेमिंग से बदल दी, CMS कहते हैं, है ना। Now the most important aspect of communication satellite, the payload, is transponder, and transponder stand for transmitter and responder. So a radio device which can receive signal, amplify it, and it can give it back to you. That is what we call it transponder. Any communication satellite's strength, capacity will be measured by the number of transponder the satellite is holding. Right? अगर 12 transponder है तो बढ़िया है, 18 है तो उससे भी बढ़िया है. Right? So we try to pack maximum transponders in one satellite nowadays. And that is why when we are sending communication satellite, we are packing 36 to 48 transponder in one satellite. Now 36 to 48 transponder that if you place in one satellite, then the weight of the satellite is going to be around 4 tons. वो 4 टन या उससे ऊपर चला जाएगा. 48 डाल दिया तो 4 टन से ऊपर. Right? And now when you have a satellite which is beyond 4 tons, तो फिर आपके पास रॉकेट नहीं है उसको लेके जाने के लिए. Right? Because our LVM-3 is a most powerful rocket and it can carry 4 tons only, up to 4 ton. अभी लास्ट के जो 2 सैटेलाइट हमने लॉन्च किए हैं वो एक था 4300 किलोग्राम यानी 4.3 टन और दूसरा था 4.7 टन. सबसे लेटेस्ट जो हमने लॉन्च किया वो क्या था 4.7 टन CMS-03 और उसको लॉन्च किससे किया? फाल्कन रॉकेट से, स्पेसएक्स अमेरिका, एलॉन मस्क. Why? Because the lift up capacity of our existing launch vehicles are not sufficient. When we developed LVM-3 in 2017 it was good enough. क्योंकि 2017 में हम 4 टन तक ही सैटेलाइट बनाते थे, टेक्नोलॉजिकल डिमांड उतनी ही तक चाहती थी. So in 2017 we were like कि now we have developed LVM-3 and India has become independent in terms of satellite launches और हम सारे सैटेलाइट लॉन्च करेंगे. But now in the last 8 years things have changed. सैटेलाइट्स भारी बनने लगे, हेवी, और हमारी लिफ्ट अप कैपेसिटी वहीं तक रह गई. दैट इज व्हाई वी आर डेवलपिंग NGLV. तो एक तो ध्यान में रखना है, इंटीग्रेशन इज वेरी इंपॉर्टेंट. यूपीएससी इज नॉट गोइंग टू आस्क यू क्वेश्चन इन वन कंपार्टमेंट वाइज, नहीं पूछेंगे, है कि नहीं? क्योंकि ऐसे पूछेगा तब तो बन ही जाएगा आराम से. तो द क्वेश्चंस वुड बी encompassing many concepts together. Sometimes the concept would be within the chapter और कई बार तो कॉन्सेप्ट ऐसे होते हैं जो आउटसाइड द चैप्टर होते हैं. जैसे कि सैटेलाइट पूछते पूछते वेब में चला गया कि वेब फिजिक्स के हिसाब से सैटेलाइट एंड द अदर थिंग दे आर आस्किंग, so we'll have to merge it together anyway. So जो ट्रांसपॉन्डर हम यूज करते हैं, जो आज के डेट में ट्रांसपॉन्डर यूज ज्यादा कर रहे हैं, तो पॉपुलर ट्रांसपॉन्डर तो उसमें एक KU बैंड ट्रांसपॉन्डर है. KU बैंड because it is being used in DTH, and KA band. KA band is being used for research and internet. Internet, in-flight इंटरनेट भी इसी से चलेगा. In-flight internet. Right. And if we go with the two very important application of remote sensing, communication. So two most important application of communication satellite. तो पहला है DTH, direct to home television, or we call it satellite television. And the second one is VSAT, very small aperture terminal. That means a small एंटीना से आप क्या करते हो, कम्युनिकेशन चैनल्स को एस्टैब्लिश करते हो. And remember, these two applications are based on KU band transponder. That is why this transponder is important. So there could be many type of transponder, S band, then C band, extended C band, KU band, K band, L1 band, L5 band. Many transponders are there. These are the numbers and digits. But हम सब याद नहीं रख पाएंगे. DTH is at KU band. K band is now being explored in research, internet, in-flight internet and all. तो लेटेस्ट जो सैटेलाइट हमने लॉन्च किए, if we go with the latest one, and just for the understanding purpose, numbers are not important. एक तो था GSAT 24. It's Indian communication satellite launched by Ariane 5, France. So Ariane 5 France, right? And this satellite is being used by entire satellite is being used by Tata Play DTH सर्विस. तो ये भी एक इंपॉर्टेंट इंफॉर्मेशन है कि टाटा स्काई नाउ इज टाटा प्ले. स्काई वाज यूके बेस्ड टेक्नोलॉजिकल पार्टनर. स्काई अब चला गया. टाटा अब अकेला इस सर्विस को मैनेज कर रहा है और सैटेलाइट उसने इसरो से ले ली. ये सैटेलाइट जो था वो डेवलप भी किया गया था टाटा के लिए, राइट. देन वी हैव लॉन्च्ड द लेटेस्ट वन. इसके बाद GSAT 20, जिसको CMS 03 भी कहते हैं. And it was launched by Falcon 9 SpaceX. Why? Because it is more than 4 tons. Like I told you, any satellite more than 4 tons, we won't be able to launch it. And this is for internet. Especially in-flight internet. So this in-flight internet is important. The term is important. Yes, in aeroplane we can have this internet. Right. Now the next one we can talk about is navigation satellite. तो इंडिया के पास 2 नेविगेशन सिस्टम है अभी. एक का नाम है नाविक, दूसरा का नाम है गगन, है ना. तो इफ वी टॉक अबाउट इंडियन navigation system, so we can divide into 2 categories. One is called SBNS, and SBNS is our Navik. SBNS is satellite based navigation system. Another one is called SBAS, that is Gagan. That is satellite based augmentation system. So there is a difference between these two. नाविक तो जानते ही आप, navigation with Indian constellation, and Gagan is GPS added. Geo augmented navigation. तो गगन हमारे पास पहले से था, नाविक हमने बाद में बनाया. And Gagan was developed just to improve the navigation signal in Indian airspace for aviation purpose. Like I told you in the classroom that all over the world, aviation has accepted GPS as a standard navigation system. All over the world, all the aviation company, they are having their aircraft equipped with GPS receivers. So pilot in the cockpit will be able to receive all the information on GPS receiver, and that will add to the security of the aircraft, right? It will also help in easy air traffic management. It will ease the congestion at the airport. So all kind of benefit would be there. But again, GPS is owned by American Defense Department, and GPS is available for civilian application all over the world just because America has opened it. अमेरिका ने इसको ओपन कर रखा है कि नॉन इंक्रिप्टेड जनरल सिग्नल सबके लिए है, जिसको यूज करना है कर लो. बट रिमेंबर वन थिंग दैट दिस जीपीएस अमेरिकन प्रॉपर्टी, you can avail it, but if quality of services are not up to mark, you cannot complain anywhere. Reason, it is just a kind of good gesture from America to open it so that it can be utilized. आप उसमें ये नहीं कर सकते कि हमारे यहाँ खराब आ रहा है तो ठीक कर दो, right? And the same thing happened in India, that in India there are some patches, and large part of the India rather, where the GPS signals are not very accurate. The inaccuracy can go up to 50 meters. स्टैंडर्ड कितना है? 3 मीटर. The standard accuracy 3 मीटर, हमारे यहाँ चला जाता है 50 मीटर तक. And if 50 meter inaccuracy is there, then we cannot use it with confidence in aviation. मतलब है भी, फिर भी यूज नहीं कर पाएंगे. वो तो आजकल गूगल मैप पे चलाना है ना कार. तो कार चलाना ठीक है, गूगल मैप मैं भी यूज करता हूँ, मैं तो दिल्ली में भी यूज करता हूँ. रीजन है कि आप नए शहर में जब आते हो, इट्स बीन ओनली 25 ईयर्स इन दिल्ली, और 25 ईयर में हाँ, तो जो लोग 3 जनरेशन से रह रहे हैं उसके हिसाब से तो ओनली है ना. अब मेरे जो फ्रेंड है, ही नोज एवरी लेन ऑफ दिल्ली एंड ही विल फ्लॉन्ट कि यहाँ से ऐसे निकल के ऐसे, मतलब कहीं अगर मान लो रात में खाना वाना खाने गए, फैमिली डिनर एंड ऑल, वो अपना कार पता नहीं किधर घुमा लेते हैं, जस्ट टू शो ऑफ दैट आई नो द शॉर्टकट्स ऑफ दिल्ली एंड आई कैन रीच टू द कॉलोनी बिफोर यू. और वो नीचे कार लगा के और आराम से टहलते रहेंगे. मैं तो 15 मिनट से टहल रहा हूँ, आप अभी आ रहे हो, कहाँ रह गए थे? मैं समझता हूँ कि क्या फ्लॉन्ट कर रहे हो. द थिंग इज दैट थर्ड जनरेशन है दिल्ली में, है कि नहीं? तो थर्ड जनरेशन है, मतलब बचपन ही दिल्ली में गुजरी है, हमारी थोड़ी ना गुजरी है. हमारे गांव चलो देखो, एक गली मुझे पता है आज भी, है ना? हाँ, तो मेरे बच्चों को पता चलेगा अब जब बाहर निकलेंगे तो दे विल बी नोइंग, राइट? लेकिन अब उनको भी पता नहीं चलेगा, क्योंकि हमारे फ्रेंड को इसलिए पता चला कि व्हेन ही वाज ड्राइविंग और बाइक और कार, तो गूगल नहीं था. अभी क्या है कि मैं भी मान लो कहीं जाता हूं तो गूगल, मतलब ये आदत हो गई है ना एक. मतलब सोचो, मुझे पटेल नगर ही आना होता है एंड आई नो द रूट, बट वो गाड़ी में बैठते ही वो मैप ओपन होता है, तो उसमें वर्क करके है, वर्क दबाना है, बस अपने आप वो लग गया. तो एटलीस्ट आई गेट एन आइडिया कि कितना टाइम लगने वाला है, आगे कंजेशन है नहीं है. तो बेसिकली दिमाग आपका जो होता है ना, वो काम करना बंद कर देता है कि अब ठीक है, रास्ता यही बताएगा. लेकिन इतना भी बंद मत करो ना कि आगे ब्रिज टूटा हुआ है, आपको दिखे ही नहीं. इतना तो दिखता ही होगा. मतलब इसका मतलब ये है कि आप गूगल को देख के चला रहे हो कि रेड होगा तभी हम ब्रेक लगाएंगे. सामने अगर कोई गाड़ी है एंड इफ द कार और बस इज स्टैटिक, इट्स नॉट मूविंग, यू गेट टू नो, राइट? आप तो ब्रेक लगाते ही हो. तो पता नहीं वो लोग फिर अच्छा ये सारे के सारे एक्सीडेंट्स हुए किधर है? यूपी, बिहार और टियर 2 टियर 3 सिटीज में होते हैं ये, जहां पे ट्रैफिक पुलिस है नहीं। और उसमें एक बहुत बड़ा मतलब उनका प्रॉपर टेस्ट पेश होना चाहिए कि इन्होंने कुछ पिया हुआ तो नहीं था, कोल्ड ड्रिंक वगैरह टाइप से, क्योंकि चांसेस आर वेरी हाई। इस तरह की नेग्लिजेंस तभी हो सकती है। बैन का मतलब सिंपल एक चीज होता है, इफ सिस्टम वांट्स टू इंक्रीज द प्राइस एंड इफ द सिस्टम इज नॉट एबल टू इंक्रीज बाय लेजिटिमेट वे, बैन कर दो, प्राइस बढ़ जाएगा, स्टेक होल्डर्स को बेनिफिट मिल जाएगी। तो आज के रेट में जो जहां पे भी बैन है वहां पे स्टेट को फायदा पहुंचेगा ना, उल्टा पहुंचेगा। कैसे नहीं पहुंचेगा? स्टेट को फायदा पहुंचता है, इलेक्शन में इतने पैसे कहां से आते हैं? आई नो वन ऑफ माई रिलेटिव इज इनटू दैट बिजनेस ओनली एंड ही इज अर्निंग, मतलब वो दिल्ली में फ्लैट रेट खरीद लिया अभी। वो बोलता है कि बैन रहे बढ़िया है, एक तो दुकान खोलने की कोई टेंशन नहीं, होम डिलीवरी कर रहा हूं मैं। और होम डिलीवरी में क्या होता है ना, वो एक हमारे माय ब्रदर्स आई मीन फ्रेंड, ही डाइड बिकॉज ऑफ ओवर ड्रिंकिंग इन बिहार व्हेन दिस इज बैन। और वो बहुत मतलब ही वाज आल्सो वंस ही वाज वेरी क्लोज टू मी टू। तो जब मैं उनके घर गया, मैंने लिवर बिल्कुल बर्बाद, और डॉक्टर ने कहा कि अब आप छोड़ दो नहीं तो मर जाओगे। तो आउट ऑफ कंसर्न, मतलब एवरीबॉडी वाज नोइंग कि ही इज नॉट गोइंग टू सर्वाइव, तो होता है ना कि चलो लेट्स, अगर आप गए हो तो मिल लो। एंड ही वाज नॉट अ बैड मैन, मतलब ऐसा नहीं कि इंसान बुरा नहीं था। तो मैंने उनसे पूछा एक सिंपल सी बात, मैंने कहा हार्डवेयर का उनका शॉप है अपना बढ़िया, बहुत रनिंग बिजनेस, काफी पैसे। तो मैंने पूछा कि भैया मतलब यू आर फाइन, आई मीन ऐसी तो कोई एडिक्शन थी नहीं आपको, तो इतना कैसे डिग्रेड हो गया? उन्होंने जो इसके ऊपर एक्सप्लेनेशन दिया ना, दैट वाज आई ओपनर। उन्होंने कहा कि देखो जब तक ये बैन नहीं था तब तक कोई दिक्कत नहीं हुई मुझे। रीजन ये था कि जब बैन नहीं था तो इट वाज इजिली अवेलेबल, आई नो, आई हैड ए कॉन्फिडेंस दैट व्हेनेवर आई फील लाइक आई कैन सेंड समवन एंड समवन कैन गो एंड बाय इट फॉर मी। और फिर हमारे फ्रेंड सर्कल भी ऐसे ही थे। शुरुआत तो ऐसी ही होती है ना कि चलो बैठते हैं, बैठते हैं का मतलब ही होता है भाई, है ना? जैसे होता है ना कि चाय पीते हैं, दूसरा एग्जाम्पल नहीं, बैठते हैं, बस हो गया, चाय कैंसिल। है ना? तो वो था कि ठीक है, उस समय ऐसा होता था कि एवरी इवनिंग 1 or 2 पीपल विल कम कि आज ठीक है बैठना है, तो हाँ ठीक है बैठ जाते हैं, एंड देन यू विल गेट इट। अब ऐसा है कि आपने एक पर्टिकुलर अमाउंट खरीदा एंड व्हेन इट गॉट फिनिश्ड आप उठ गए, खत्म, राइट? ऑल्दो इट्स नॉट गुड, इट्स नॉट जस्टिफाइड, बट फिर ठीक है, मतलब कम से कम लिवर डैमेज तो नहीं हो रहा। अभी क्या, कोविड, एंड कोविड के साथ साथ एक और प्रॉब्लम, बंद हो गया सब कुछ। तो हुआ क्या कि वो सारे फ्रेंड सर्कल डिस्पर्ज हो गए। नाउ द शॉपकीपर हु इज सेलिंग दिस लिकर, ही इज कॉलिंग एवरीवन, इफ यू वांट आई कैन गिव सप्लाई टू यू। अब क्या है ना, सप्लाई वो डिले नहीं देने आएगा ना, वो कहता है कि एक बार में आप ले लो, स्टॉक रख लो। तो उन्होंने कहा कि अब क्या है ना, इन माई रूम देर इज वन कॉर्नर, एक वहां पे रखा हुआ है, स्टॉक आता है, वहां पे स्टॉक आता है, तो अब तो रूम में ही है ना, और फ्रेंड्स भी शाम में नहीं मिलने वाले हैं, तो मैं सुबह उठता हूं, मेरा ध्यान चला जाता है तो मैं पी लेता हूं, फिर दोपहर में भी ध्यान चला जाता है, फिर मैं पी लेता हूं, फिर शाम में भी घर में ही अवेलेबल है अब तो फ्रीक्वेंसी ऑफ ड्रिंकिंग दैट, वो पहले वंस इन अ डे था, अब वो थ्राइस इन अ डे हो गया। एंड दैट इज हाउ, पैसे की कमी तो है नहीं, है कि नहीं? इस चीज के लिए भी पैसे की कमी होती भी नहीं है, लेकिन प्रॉब्लम, ही एक्सप्लेन इट प्रॉपरली, बोले कि मैं क्या करूं, अब आई एम एडिक्टेड टू इट बिकॉज इट्स देयर। मैंने कहा मंगवाओ ही मत, बोला कि मैंने कई बार कोशिश भी की, लेकिन उसमें कई बार वही कॉल कर देता है, हाँ, मना नहीं कर पाता। मतलब मुझे तो लगता है कि इस पूरे सिस्टम में जो डेडिकेशन है लोगों का, मतलब द काइंड ऑफ पैशन एंड डेडिकेशन दे आर हैविंग फॉर दीज थिंग्स, अगर इसका 1% भी आ जाए किसी और चीज में तो फिर वो माली हो जाएगा। बट द प्रॉब्लम इज दैट द सेम थिंग, द पीपल हु आर रनिंग दिस बिजनेस, मतलब होता है ना, जो इंसान जो मैं कह रहा हूं कि मेरे रिलेटिव हैं जो ये काम करते हैं, ही इज टीटोटलर, ही डजंट ड्रिंक अ सिंगल ड्रॉप। उसने कितने को मारा होगा पता नहीं, लेकिन वो नहीं, वो बस पैसे कमा रहा है उससे, दैट्स इट। वही बात हो गई ना, इलॉन मस्क से पूछा अभी हाल फिलहाल में कि आप अपने मोबाइल का स्क्रीन टाइम बताओ। एक इंटरव्यू में इलॉन मस्क से पूछा व्हाट इज योर स्क्रीन टाइम ऑफ द मोबाइल, तो इलॉन मस्क वाज लिटिल हेजिटेंट एंड ही वाज लाइक कि nowadays it's 1 hour in a day। तो वो इंटरव्यूअर परेशान हो गया कि ओनली 1 hour, मतलब इलॉन मस्क जिसने ये सारा इंटरनेट की दुनिया बना रखी है, और 1 hour। तो उसने कहा कि नहीं नहीं, आई कैन बिलीव इट, कैन यू शो मी योर मोबाइल फोन विद स्क्रीन टाइम। इलॉन मस्क वाज अगेन हेजिटेंट, ही टुक आउट द मोबाइल फोन और वो अंदर चेक वेक करके फिर वो मुस्कुराने लगा, ऐसे छुपाने लगा, फिर बोला शो मी, तो बोला कि दिस इज अ न्यू मोबाइल फोन सो इट्स ओनली 10 minutes, ओनली 10 minutes। तो उसका स्क्रीन टाइम है ओनली 10 minutes, है ना, एवरेज। वो छुपा इसलिए रहा था कि ज्यादा था, वो छुपा इसलिए रहा था कि मैं किस मुंह से 10 मिनट दिखाऊं, क्योंकि मैं ही तो दुनिया भर को ये स्टारलिंक दे रहा हूं अभी। So that is how the things happen. Anyway, so this is GPS added because this is a way by which we can improve the GPS signals. तो हमने क्या किया, हमने बीच का रास्ता निकाल लिया, satellite based augmentation system। In this system what we can do, that we can add some additional data into the GPS, or GPS data can be added to this system and overall accuracy will increase। तो इसमें क्या करना है, इसमें सबसे इंपॉर्टेंट फीचर क्या है, there is no dedicated satellite of Gagan। You never see Gagan satellite, right? Rather payload, Gagan payload, rather you can write Gagan payload, Gagan payload is payloads are placed in communication satellite, so very low cost way out। So Gagan payloads are placed in communication satellite, and the whole purpose is that GPS plus Gagan, and then it is improve navigation signals for aviation, especially for aviation। So we have it, and it was developed by Airport Authority of India and ISRO। So development, Airport Authority of India plus ISRO, Airport Authority of India and ISRO, right? So this is your SBS augmentation system, because there is no satellite launch, rather you just place the payload into the communication satellite. We are maintaining 3 communication satellites with Gagan payload, and this payload will add some signal with the GPS and all things will be done. So it's a way out to improve it. The second thing we developed was Navik, and Navik is for our own satellite navigation system. That is why it is called satellite based navigation system। So we have satellite for it, है ना। तो नाविक का जो सैटेलाइट है वही मेन इंपॉर्टेंट पॉइंट है अभी, नाविक सैटेलाइट। एंड देयर वी हैव 2 टाइप ऑफ सैटेलाइट, generation 1 that is called IRNSS, and generation 2 satellite that is called NVS satellite, right? So Navik, we all know, it's a regional navigation system of India. In this regional navigation system, the minimum satellite we need to have is 7. Out of 7 satellite, 3 should be there in geostationary orbit and 4 should be there in geosynchronous orbit, that we know. And we have established our Navik system with the IRNSS, but IRNSS is generation 1 satellite, this is advanced satellite. IRNSS क्या है ना, ये इसका वजन कम है, it's lightweight. Rather, we had to keep it lightweight because when we planned IRNSS, then we had only one launch vehicle and it was PSLV. तो ये 2014 के पहले की प्लानिंग है. So obviously we wanted to launch this IRNSS with PSLV only, and PSLV cannot lift up heavy satellite to the GSO. So lightweight, and it was launched by PSLV. और lightweight में compromise कहाँ हुआ? Transponder के साथ. That is why you know that Navik is there, it's active one, but we are not using Navik in our day-to-day life. आज भी डे टू डे लाइफ में हम जब यूज करते हैं तो हमारे पास ग्लोनेस है, ग्लोनेस है और जीपीएस है, ये यूज करते हैं. नाविक तो यूज नहीं कर रहे. तो नाविक इज ओनली बीइंग यूज्ड इन स्ट्रेटेजिक एंड डिफेंस रिलेटेड थिंग, क्योंकि प्राइम टारगेट वही था. इंडिया वांटेड टू हैव अ नेविगेशन सिस्टम so that Indian defense could rely on homegrown system, because you cannot rely on GPS. हाँ, जीपीएस पे रिलाई आप करो. जैसे मान लो हमने एक ड्रोन बनाया जिसका नाम है नागास्त्रा, हम लोग पढ़ेंगे अभी रिसेंटली. और नागास्त्रा जो है वो जीपीएस से अपना नेविगेशन सिग्नल लेता है और फिर जाके किसी भी टारगेट पे जाके वो गिर जाता है. इट्स अ सुसाइड ड्रोन, right? This drone is not going to throw some bomb, rather the drone itself will fall onto the target, drone is gone and entire thing will be, वो सुसाइड मिशन पे जाता है. तो उसमें जीपीएस कैसे यूज कर रहे हैं? ठीक है, जीपीएस यूज कर सकते हैं, बिकॉज वी नो वन थिंग ना, दैट वी आर हैविंग आवर ओन सिस्टम. ये हमारा सिस्टम है, क्योंकि वो छोटा डिवाइस है, उसमें छोटे सेंसर लगेंगे. छोटे सेंसर क्या होंगे? जीपीएस के होते हैं. क्यों होते हैं? क्योंकि जीपीएस के पास एक स्पेशल बैंड है, L1 बैंड. If you have L1 band, L5 band, then you can receive the GPS signals into watches, in handheld devices, in a small trackers. इसमें नहीं है. You know that satellite की जो weight है, it largely depend upon what? Transponders, right? So the difference between these two, जो IRNSS और NVS है, it's a lightweight. And lightweight क्यों है? Because it has got only 2 transponder, एक S band and L5 band. इसमें पैराशूट इसलिए कि कभी अगर आपको लास्ट मोमेंट में आपने कहा कि नहीं, आई विल नॉट फॉलो टू द टारगेट, तो वापस बुला के हम उतार सके, तो रिकवरी के लिए, राइट? आई मीन एवरी टाइम व्हेन वी लॉन्च नागास्त्रा क्या करेगा? वो सीख भी तो करता है ना, टारगेट कहाँ है. मान लो वहां टारगेट है ही नहीं, तो क्यों गिरा के आएंगे हम? तो विल ब्रिंग इट बैक एंड देन वी कैन रिकवर इट बाय ओपनिंग द पैराशूट. तो पैराशूट से जब गिरेगा तो रिकवर होकर दोबारा यूज हो सकता है, अदरवाइज वो फिर खराब हो जाता है. एनीवे, तो हियर वी हैव S1 एंड L5 बैंड. इससे हमारा नॉर्मल कॉमन नेविगेशन तो फुलफिल हो जाएगा, but we won't be able to use Navik in watch, in mobile phone, in handheld devices, in a small trackers. You get my point? तो हम उसी को फिक्स कर रहे हैं. ये क्या होगा? दिस इज अ लिटिल, ये हेवी सैटेलाइट है, हेवी सैटेलाइट, एंड बिकॉज इट इज हेवी, इट विल बी लॉन्च बाय GSLV Mark 2. GSLV Mark 2 से लॉन्च किया हमने. और हेवी इसलिए है क्योंकि इसमें एडिशनल S बैंड तो है ही, L5 बैंड भी है, एंड एडिशनल वी आर हैविंग L1 बैंड. L1 band, that's the most important feature. And once you have L1 band, then with this L1 band you can give signals to small wearable trackers, devices and sensors. तो आप लिख सकते हैं L1 band will provide, L1 band will provide navigation services to navigation signals to small wearable devices such as mobile phone, smartwatch, fitness tracker, and small sensors. Right. So now, once we replace all of this IRNSS with NVS, तब जाके हम इसका इस्तेमाल वैसे ही कर पाएंगे जैसे जीपीएस ट्रैकर्स का इस्तेमाल हम करते हैं. That is why in Indian market also, if you go with any navigation signals जो घड़ी वड़ी में वो भी जीपीएस यूज करते हैं. अब हम उसको लेके आ रहे हैं. तो that's the difference. That is why because you have added one more transponder, the weight will be higher. है ना. अभी प्रॉब्लम क्या आई इसमें रिसेंटली क्या आया कि दिस 7 इज देयर, यहाँ पे 7 सैटेलाइट है, 9 सैटेलाइट लॉन्च किए, 7 एक्टिव है. तो आईआरएनएस तो भूल जाओ, ये तो पुरानी बात हो गई. एनवीएस में 2 लॉन्च किए हमने, एनवीएस 01, एनवीएस 02. 01 वाज प्रॉपर लॉन्च, 02 का जो लॉन्चिंग था वो गड़बड़ हो गया. और गड़बड़ किस लिए हुआ? बिकॉज उसमें एक वॉल्व खराब था. So few terms that came very, important around that time. और एक पॉइंट लिख लीजिए, या लिखा भी होगा शायद, लिख लीजिए, दोबारा लिख लीजिए. लिखा हुआ है ना 100 लॉन्च वाला. हाँ, तो मैं समझा देता हूं फिर. तो उसमें क्या था कि NVS 02 was very landmark launch. तो NVS 02 was landmark launch because it was 100th launch of ISRO. तो ISRO ने इसको सेलिब्रेट करना चाहता कि इट इज गोइंग टू बी सेंचुरी ऑफ लॉन्चेस इन टर्म्स ऑफ सैटेलाइट्स. मतलब हम साउंडिंग रॉकेट को छोड़ दे तो ये 100 लॉन्च था हमारा. राइट. एंड इट वाज बाय GSLV Mark II. एवरीथिंग वेंट वेल, बट देन in satellite a valve stopped working, and this valve was the valve of a motor. उस मोटर में valve का काम क्या था? ऑक्सीडाइजर और फ्यूल को मिक्स करना. Because I have already explained one thing to you that when you, this is Earth, और जब आप सैटेलाइट को लॉन्च करते हो तो सैटेलाइट इज नॉट लॉन्च इनटू द ऑर्बिट डायरेक्टली, रेदर यू लॉन्च द सैटेलाइट इन GTO, और यहां से सैटेलाइट घूम घूम के आगे जाके फिर पहुंचता है. तो सैटेलाइट तो यही लॉन्च हुआ, पहुंचाना यहां है. तो सैटेलाइट यहां से यहां तक तो खुद जाएगा, एंड फॉर दैट सैटेलाइट हैज टू बर्न इट्स ओन फ्यूल. तो सैटेलाइट विल हैव 2 कंपार्टमेंट, वन इज फ्यूल कंपार्टमेंट, अनदर इज ऑक्सीडाइजर, बिकॉज देयर इज नो ऑक्सीजन, एंड फ्यूल एंड ऑक्सीडाइजर शुड मिक्स ओनली एट द टाइम ऑफ बर्निंग. अगर पहले ही मिल जाएगा तो ऑक्सीडाइज होकर खत्म हो जाएगा, ब्लास्ट कर जाएगा, है ना. तो अब क्या है उसमें कि एक वॉल्व लगा हुआ है, एक वॉल्व जो फ्यूल को रिलीज करता है, दूसरा वॉल्व. एवरीथिंग वाज फाइन, ओनली द वॉल्व ऑफ ऑक्सीडाइजर बिकम फॉल्टी, एंड नाउ ऑक्सीडाइजर वाज नॉट बीइंग रिलीज्ड, फ्यूल विल नॉट बर्न, और वो इधर ही घूम रहा है. बल्कि घूमेगा क्या, एक पॉइंट पे आके वो गिर जाएगा, खत्म. तो 100th लॉन्च, द मिशन लॉन्च इज ओके, लॉन्च तो ठीक ही ठाक था, लेकिन मिशन फेल कर गया. राइट. तो उसमें आप ध्यान रखिएगा, जो वॉल्व खराब हुआ, मोटर, एपोजी पेरेजी पढ़ते हैं आप, है कि नहीं. तो लिक्विड एपोजी मोटर, कई बार होता है ना, यूपीएससी टर्म निकाल के दे देंगे, लिक्विड एपोजी मोटर. अब बताओ कहां यूज होगा. तो बस सैटेलाइट में आप देख लो, स्पेस टेक का कौन सा ऑप्शन है उधर. तो लिक्विड एपोजी मोटर या पेरेजी मोटर, कुछ भी आ जाए. तो एपोजी पेरेजी, दैट्स ऑल्सो, जब हम पढ़ते हैं ज्योग्राफी में ऑर्बिट्स एंड ऑल, वी टॉक अबाउट इट, बट हियर इज ऑर्बिट ऑल्सो, यहां भी तो ऑर्बिट ही है. सो प्रॉब्लम क्या थी, कहां थी? सो दिस वाज फेलियर, सैटेलाइट फेल्ड ड्यू टू faulty valve of liquid apogee motor. और लिक्विड एपोजी मोटर का अगर फॉल्टी वॉल्व है तो व्हाट विल हैपन? फ्यूल एंड ऑक्सीडाइजर will not meet properly. So now the satellite is there in our GTO only. It won't be able to reach to the designated orbit, and then it will creating problem. Right. Quickly we can go with the weather satellite. So we are having 3 weather satellites which are very good. INSAT 3D, INSAT 3D-R and INSAT 3D-S. So these all are advanced weather satellite of India. जो लेटेस्ट हमने लॉन्च किया है वो 3DS है, that was launched in the last year only. So 2024. Right. Now these weather satellites are advanced one because it's a world class. Second thing, that this satellite is important because this satellite is also holding special payload for search and rescue. तो एक पॉइंट लिख लीजिए. These satellites are also having search and rescue payload. Search and rescue payload, and India is part of international international satellite based search and rescue system. Search and rescue system, right? Besides, the satellite will have what? Sounder and imager as payload. तो इसका जो normal payload होगा तो जिसको weather payload कह सकते हैं. So weather payload or meteorological payload is sounder and imager. Imager is nothing, it will click the photograph of entire India and the area around to get the location of the clouds and everything. Sounder will give you, sounder is important one, that will give you vertical profile of temperature and humidity. Vertical profile of temperature and humidity. तो इनसेट 3D मीडियम वेट का है, लॉन्च हम इसका करते हैं GSLV Mark II से. So the last one was launched by GSLV Mark II. Right. Okay. Now, if we go with another one, the fifth area, then there is something called astronomical satellite. Astronomical satellite we are having 3 now. एक है एस्ट्रोसैट, फिर दूसरा आदित्य L1, एंड थर्ड वन इज एक्सपोसैट. और अभी फिलहाल एस्ट्रोसैट 2 की बात भी होने लगी है. ऑब्वियसली जाएगा ही. Right. So astrosat was in 2015. That's not important, but again it was first astronomical satellite. Or there is another word for it that is called a space observatory. स्पेस ऑब्जर्वेटरी. एस्ट्रोसैट ने कुछ खास कमाल नहीं किया, बट ठीक है. It's a first space observatory. That means we have placed 5 telescopes in a space, in orbit basically. तो 5 टेलीस्कोप ऑर्बिट में हमने पहुंचाया. Small telescope, not very advanced one. It's a normal one only. Right. These 5 telescopes will work in range of visible light, UV rays and X-ray. So visible light, UV rays and X-rays. So one thing you should be very clear about, the logic and reason behind placing the telescope in space. I mean, we can make much bigger and better telescope on Earth, and we can establish here also. Rather, we have a telescope like that. India has got good telescopes, I mean powerful telescopes on land. जिसको आप सकते हैं कि हमारे जो स्पेस ऑब्जर्वेटरी लैंड पर है. तो मैंने क्लास में बताया भी था कि जो भी सेलेस्टियल ऑब्जेक्ट हैं, लेट्स से स्टार, नेबुला, any kind of galaxy or any planet, so how do we study them? Again we do remote sensing. उनका भी तो remote sensing ही करते हैं या स्पेक्ट्रोस्कोपी करते हैं. So we get all the information about celestial object by the waves which are coming from those objects. हम कहते हैं दिखता है, दिखता नहीं है. दिखने का कॉन्सेप्ट भी वही है कि the light coming from that object must enter in your eye. It must make an image on a retina. Then only you see that आप देख रहे हैं. हमें कई बार लगता है कि देखना एक्टिव प्रोसेस है. नो, इट्स अ पैसिव प्रोसेस, है ना. Because if the light will enter, fall onto the retina, image will form, then only you will be able to see. तो अगर हम स्टार्स को भी देख रहे हैं ना, तो पहली बात कि वो स्टार को इसलिए देख पा रहे हैं because the light from that star is able to enter in our eye, right? लेकिन क्या है ना, अभी पढ़ा आयनोस्फेयर आपने. देयर इज मैग्नेटोस्फेयर, देयर इज ओजोन लेयर, देयर इज अ लॉट मेनी लेयर्स व्हिच आर बेसिकली शील्ड ऑफ द अर्थ. एंड दिस शील्ड्स, दिस आयनोस्फेयर, मैग्नेटोस्फेयर, ozone layer, stratosphere, these shields will not allow many radiations of the space to enter into the earth, and that's very good for the life. नहीं तो गामा रेज भी आ जाता, एक्स रेज भी आ जाते हैं, यूवी रेज सी भी आ जाता, और हम सब होते ही नहीं, कभी लाइफ ही ओरिजिनेट नहीं होता. टू ओरिजिनेट लाइफ ऑन एनी प्लैनेट, दिस इन्वायरमेंटल शील्ड मस्ट बी देयर. इसलिए जहां पे भी एटमॉस्फेयर नहीं है वहां पे बात ही नहीं होती कि कुछ हो सकता है. But then at the same time, for telescope which is there on ground, these all waves are not available to them. तो फिर वो limited हमें information देता है. That is why Hubble telescope and James Webb telescope, they are able to give you much more detailed images and information about the celestial objects and celestial phenomena, because they are getting all these X-rays को भी वो ले रहे हैं अच्छे तरीके से. हालांकि ऐसा नहीं है हम ग्राउंड पे भी जहां भी हम टेलीस्कोप लगाते हैं वहां पे भी हम गामा सेंसर्स लगाते हैं, because traces of gamma radiation would be coming, traces of X-ray would be coming, but then it's a traces because all of them, 99% of them are being filtered by the atmospheric protective shields. So the purpose of putting this telescope outside the atmosphere is to expose the sensors and telescopes to gamma radiation, X-rays and ultraviolet rays. जो जितनी अधिक रेज मिलेगी उतना ही इंफॉर्मेशन मिलेगा, है कि नहीं? So that is why everybody and every space superpower, they are willing to place it to that particular height, है ना? तो एक लॉजिक याद रखना, in that logic they can explore something because obviously UPSC, I mean, मैं जब कहता हूं कि नहीं पूछेगा इसका मतलब नहीं पूछेगा, नहीं पूछ तो सकते ही है वो, फैक्चुअल पूछ सकते हैं, लेकिन फैक्चुअल कितने पूछेंगे आप सोच के देखो. आउट ऑफ 100 क्वेश्चन दे आर गोइंग टू आस्क 20 क्वेश्चंस व्हिच आर फैक्चुअल, मतलब फैक्चुअल और नहीं फैक्चुअल, और वो डिसाइसिव तो नहीं हो सकता ना. दोज फैक्चुअल और नियर फैक्चुअल क्वेश्चंस विल नॉट डिसाइड कि सिलेक्शन होगा कि नहीं होगा, क्योंकि वो सब बनाएंगे. हम जब पढ़ते हैं तो उतना पढ़ लेते हैं कि वो बन जाएंगे. तो द क्वेश्चंस व्हिच विल डिसाइड, इट वोंट बी फैक्चुअल. अच्छा, फैक्चुअल में भी दो तरीके हो सकते हैं, ऐसा कि कुछ ऐसा ही डेटा पूछा कि आता ही नहीं किसी को, तो किसी को नहीं आता तो ठीक है ना, किसी को नहीं आएगा. तो डिजिट्स, आई मीन इफ यू गो थ्रू द पास्ट ईयर क्वेश्चन पेपर यू विल फाइंड द डिजिट्स नहीं पूछ रहे. एंड इवन इफ दे आस्क सम डिजिट्स देन इट्स वेरी हार्ड टू क्रैक दैट क्वेश्चन. मतलब वो बहुत कोइंसिडेंस वाली बात होती है कि आपको वो डिजिट याद है. दैट इज व्हाई वी शुड नॉट put so much of focus on numbers. Here could be also factual, but then every fact must have some conceptual clarity. It's not like कि वहां पे है, वहां पे क्यों है ये भी इंपॉर्टेंट है ना. Why these space observatories are there in a space? What kind of benefit we are going to get? It's just like it is outside the protective field or protective shield of the atmosphere, so that it can get exposed to all kind of radiations which are coming from celestial bodies. More radiation, then more information. Because whatever we know about those stars and celestial body is based on what we are receiving on this earth. Rather, कई बार ये भी आता है ना कि हम जितने स्टार्स देखते हैं, जितने स्टार्स हम देख पा रहे हैं, उसमें से आधा से ज्यादा तो है भी नहीं, उनकी लाइट ही आ रही है अब तक, है कि नहीं? क्योंकि वो इतने लाइट ईयर दूर है, पता चला कि वो 1 आध मिलियन लाइट ईयर दूर है, तो वो खत्म हो चुके हैं, अभी वहां कुछ नहीं है. इट्स जस्ट लाइक लाइट इज स्टिल कमिंग, क्योंकि लाइट निकल चुका है तो वो आ रहा है अभी भी. बट ठीक है, इन दैट वे अगर आप थोड़ा सा जाओगे ना, मैंने एक बुक मिली थी मुझे, मैं पढ़ रहा था तो लगा, कई बार तो ऐसा लगता है कि वाई वी आर डूइंग ऑल दिस थिंग्स देन इफ इट्स नॉट देयर, राइट. तो फिर ठीक है, उसमें एक आता है कि ह्यूमन क्यूरियोसिटी, इफ यू वोंट डू इट तो फिर नियोलिथिक पैलियोलिथिक टाइप रहेगा मामला. So we need to engage into something, right? That is called recreation or hobby or whatever. Anyway, Aditya L1 is again important one because ये इंडिया का सेकेंड मिशन हुआ एंड फर्स्ट मिशन फॉर स्टडी ऑफ सन. Here the important thing is L1 itself. L1 is Lagrangian point 1. And Lagrangian point 1, Lagrangian points are those points where the net gravity will be 0. Anything placed around this point will move around that point only. That is why Lagrangian points where nothing is there but still satellite will move around. यहाँ है कुछ भी नहीं, ये पॉइंट है जहां पे कुछ भी नहीं है, सैटेलाइट इसी के चारों तरफ घूम रहा है. नॉर्मली सैटेलाइट घूमता है किसमें? ग्रेविटी फील्ड में. यहाँ कुछ भी नहीं है फिर भी घूम रहा है, बिकॉज यहाँ नेट ग्रेविटी 0. So that is why this area is also known as parking space in space. Parking space in space. So whenever the term parking space comes in that context, it simply means they are referring to Lagrangian point. So L1, L2, L3, L4, L5. L1 पे हमारा है, and for that way you need to understand L2 पे जेम्स वेब है. तो अमेरिका का जेम्स वेब टेलीस्कोप किधर है? L2 पे. Although Aditya L1 is not as powerful as James Webb, for that matter James Webb का पर्पस ही कुछ और है. This is only for the study of sun, and we have placed it at L1 point, which is 15 lakh kilometer, 1.5 million kilometer away from Earth. One of the benefit that L1 can give you, that it is always exposed to the solar radiation. हमेशा सोलर रेडिएशन से एक्सपोज होगा. It never come to the dark side of the earth because it is far away from the earth, not moving around the earth, rather it is moving around the sun, जिसको हेलो ऑर्बिट या हेलियोसेंट्रिक ऑर्बिट कहते हैं. क्योंकि मामला ऐसा है कि ये सन है, ये अर्थ चारों तरफ घूम रहा है और ये L1 पॉइंट है. So this entire system will move, this line will move. So this line will keep on moving. So L1 is always exposed to the sun. Moon is here, तो मून से भी कोई प्रॉब्लम नहीं है, इक्लिप्स का भी कोई इंपैक्ट नहीं होगा. तो ऐसा कुछ नहीं है, इट्स अ काइंड ऑफ टेक्नोलॉजिकल डेमोन्स्ट्रेशन. लाइक आई टोल्ड यू इन द क्लास ऑल्सो कि ओरिजिनल प्लान L1 था ही नहीं. ओरिजिनल प्लान था सिंपल आदित्य बाय PSLV एट 800 किलोमीटर पोलर ऑर्बिट, LEO में. लेकिन वही होता है ना, समय बीच से चला जाता है, हम कर नहीं पाते. देन हम मिशन ऑब्जेक्टिव्स को हम क्या करते हैं, रिवाइज करते हैं, उसके स्कोप को बढ़ा देते हैं. तो वी kept on adding a scope of the mission objectives, and then we realized कि अभी क्या ये 800 किलोमीटर पे भेजना है, that we can do easily. So let's do one thing, that place it at L1, जहां पे अभी तक बहुत कम कंट्रीज, जो डेवलप्ड कंट्री है उसमें से 2-3 पहुंचे, सभी भी नहीं पहुंच पाए. So that is technology demonstration to put it over there. Next one is your XPOSAT. XPOSAT is X-ray polarimeter satellite. एस्ट्रोसैट और एक्सपोसैट में एक छोटा सा अंतर है. एक्सपोसैट वाज सेंसिटिव टू विजिबल लाइट, एक्सरे एंड यूवी रेज. दिस इज ओनली फॉर एक्सरे, एंड नॉट ओनली एक्सरे बट एक्सरे प्लस पोलराइजेशन एंगल ऑफ द एक्सरे. ये दो को एक्सप्लोर कर रहा है. एक्सरे, एक्सरे में दो चीज, मतलब फ्रीक्वेंसी एंड polarization angle, frequency and polarization angle. So in wave it's not only frequency which is important, obviously the most important thing is frequency only, but polarization angle is again something else. polarization मैंने बताया था शायद आपको कि जैसे मान लो हम क्वांटम फिजिक्स में क्या पढ़ते हैं कि every EM wave is made up of a small particle and that particle is called photons. लाइट के पार्टिकल को फोटोन्स कहते हैं. तो लाइट भी अगर ट्रेवल हो रही है तो क्वांटम फिजिक्स कहता है कि लाइट की ट्रेवलिंग, इट्स नॉट ओनली एनर्जी एंड वेव, इट्स अ पार्टिकल आल्सो. रादर क्लासिकल फिजिक्स ने क्या कहा कि लाइट इज नथिंग बट द एनर्जी फ्लो. क्वांटम फिजिक्स ने कहा कि ठीक है एनर्जी तो है लेकिन पैकेट के फॉर्म में, और वो पैकेट फोटोन है. इट्स नॉट only energy is there but particulate nature में भी है. It's a particle which is carrying energy. That is why there is always ongoing conflict कि जो लाइट है वो पार्टिकल नेचर का है या वेव नेचर का है ना. तो अगर पार्टिकल है, और अगर क्या है ही पार्टिकल है ना, तो पार्टिकल की मूवमेंट ऐसे से भी हो सकती है, क्योंकि मान लो यही पार्टिकल है और ये मूवमेंट इस एंगल पे भी हो सकता है, एंगल बदल गया. तो ये पोलराइजेशन एंगल है, राइट. दिस एंगल, दिस एंगल, दिस एंगल, कुछ भी हो सकता है. तो पोलराइजेशन एंगल इज लाइक दिस. सो इफ देयर इज अ पार्टिकल, ये तो ज्यादा बड़ा पार्टिकल, मान लो ये पार्टिकल है, दिस इज पैकेट, सो दिस पैकेट कैन मूव लाइक दिस, ऐसे करके आ रहा है. तो ये पार्टिकल ऐसे से भी तो मूव करता हुआ आ सकता है कि नहीं. फ्रीक्वेंसी भी वही रहेगी, डायरेक्शन भी वही रहेगा, बस पोलराइजेशन एंगल चेंज हो गया, एंड दैट विल गिव यू एडिशनल इंफॉर्मेशन, राइट. मैंने तभी आपको शायद बताया था कि पोलराइजेशन, अब होता क्या है ना, पोलराइजेशन एंगल, पोलराइजेशन, दिस ऑल बिकम इंपॉर्टेंट. तो यू शुड बी नोइंग एट द सेम टाइम कि हम पोलराइजेशन का यूज करते हैं, है कि नहीं. तो एक करते हैं कहां पे हम 3D मूवीज में. 3D movies are shot by two camera and these two cameras are basically shooting it at two polarizations of light. तो दो पोलराइजेशन ऑफ लाइट पे शूट करती हैं, एक्स और वाई पे, है कि नहीं. मान लो एक्स एंड वाई टू पोलराइजेशन पे शूट कर रहा है. फिर हमें चश्मा देते हैं वो, है कि नहीं. क्योंकि अगर आप बिना चश्मे के देखोगे तो एक्स वाला भी आएगा, वाई वाला भी आएगा और दोनों आंखों में जाएगा, तो ब्लर दिखेगा, दो इमेज दिखेंगी. तो पोलराइजेशन पे पर्दे पे जो इमेज है उसमें एक इमेज है एक्स पे, दूसरा वाई पे. तो इधर वाले चश्मे पे क्या लगा है, एक फिल्टर जो सिर्फ एक्स को अंदर जाने देता है, वाई को रोक देता है. तो एक आंख एक इमेज देखेगा और दूसरा वाला दूसरा देखेगा, क्योंकि ये सिर्फ वाई को जाने देता है, एक्स को रोक देता है. तो टू इमेज by two eyes superimpose and then it will give you the depth effect. क्योंकि हम भी अपनी दो आंखों से देखते हैं ना, दो इमेज दिखती हैं. आपको दो नहीं दिख रहा होगा अभी, लेकिन एक आंख बंद करोगे तो एक इमेज दिखेगा और दूसरी आंख बंद करोगे तो थोड़ा सा उसके साइड में दूसरा दिखता है ना. छोटे बच्चों को देखा कभी आपने, एक एक आंख बंद करके ऐसे ही खेला होगा. जिस दिन ये पता चलता है ना कि दो आंखों से दो इमेज दिखता है और वो ऐसे ऐसे आगे पीछे होता है, तो हम कुछ दिन तक तो इसी को मजे लेते हैं, इसी में. लेट्स मूव द इमेज, इधर से इधर, इधर से इधर, इधर से इधर. तो ब्रेन टू इमेज को परसीव किया, सुपर इम्पोज किया ब्रेन, एंड देन ब्रेन गॉट द सेंस ऑफ डेप्थ. अब सेम हमें सेंस ऑफ डेप्थ देना है, ब्रेन को डिसीव करना है. तो डिसीव कैसे किया, तो पोलराइजेशन, लेट्स आंखों पे लगाओ फिल्टर, एक से एक जाएगा, दूसरे से दूसरा जाएगा, सेम सिग्नल ब्रेन को मिलेगा कि अच्छा दो आंखें दो चीज देख रही हैं. और फिर वो डेप्थ, हालांकि वो देख आप 2D पे रहे हो, 2D प्लेन चीजों थोड़े ना दो दिख सकता है. तो दैट वी गेट द डेप्थ. सेकंड पोलराइजेशन कहां पे होता है, जो एंटी ग्लेयर ग्लासेस होती है. एंटी ग्लेयर ग्लासेस आप जाते हैं लेंस कार्ड में देते हैं, ये वाला देखो, इसमें आप रात में गाड़ी चलाओगे तो फ्लेयर्स नहीं दिखेंगे, वो भी पोलराइजेशन का ही मामला है. वो कुछ रखते हैं ना, देखेंगे, देखो यहाँ पे देखो दिख रहा है ना, अब ये चश्मा लगा के देखो, आपको एकदम क्लीन दिखेगा. तो दैट इज ऑल्सो वर्किंग ऑन पोलराइजेशन, है ना. तो दिस एप्लीकेशन शुड ऑल्सो बी देयर टू लिंक इट, है ना. एस्ट्रोसैट अभी आया है, वी डोंट नो द प्रॉपर स्पेसिफिकेशन ऑफ एस्ट्रोसैट कि व्हाट एल्स कुड बी देयर. यस, मे बी विद एस्ट्रोसैट 2 हम लोग इसमें और बेहतर कुछ करने वाले हैं, है ना. तो आज इतना ही रहने देते हैं, 2 बजे तक चलता है चलाने को. हाँ बहुत है, तो कल मिलेंगे, कल खत्म ही हो जाएगा. तो वी आर लेफ्ट विद आईटी. नहीं मंडे को कहा, 2 दिन बहुत है. हाँ हाँ संडे को है. मुझे तो संडे को बुलाया है 8 से. हाँ क्लास है, क्या बात कर रहे हैं. हो जाएगा, अपडेट हो जाएगा, अभी करवा देता हूँ. कोई नहीं, आ जाना, है ना. रिवीजन ही हो जाएगा ना, इट्स अ काइंड ऑफ रिवीजन ओनली. टेस्ट दे देना बाद में, कोई बात नहीं.